Good afternoon, ladies and gentlemen. We warmly welcome you all to the 14th International Research Conference of the General Sir John Kotelawala Defence University. Thank you very much for joining with us physically as well as virtually today. We are about to begin the plenary session of the Basic and Applied Sciences track under the theme Holistic Growth and National Development. Today, we have 5 eminent international research scholars lined up for you, and the session will be chaired by 2 renowned professors. Let me now cordially invite our Basic and Applied Sciences track chairperson, Professor Charitha Gunasekara, to introduce the session chairs. Professor Gunasekara is the current Dean of the Faculty of Research and Development and the Director of the Institute for Combinatorial Advanced Research and Education, KDU-CARE. Over to you, Madam.
Thank you, Dr. Angela. It is my honor to introduce the co-chairpersons of the plenary session in Basic and Applied Sciences, Professor Ranil D. Silva and Professor George Dias. So let me introduce first Professor Ranil D. Silva. Professor Ranil D. Silva is a research professor attached to KDU-CARE. His area of research interests include healthy brain aging, genetics, and fluid biomarker of neuromuscular and neurodegenerative diseases leading to personalized medicine. He became the spearhead in establishing 2 of the largest biobanks in South Asia, a brain bank with the most comprehensive neuropathology, and a DNA bank of neurological disorders inclusive of rare diseases. Serving to the nation, he provides, actually his laboratory provides, free biomolecular diagnostics for neurogenetic diseases. He has been the principal supervisor for several double doctoral and master's degrees in neuroscience with prestigious international universities. He is a member of the Governing Council of International Brain Research Organization, France Council member of the Federation of Asian and Oceanic Neuroscience Societies, Japan and Asian Pacific Society for Neurochemistry, Singapore, Fellow of the Indian Academy of Neurosciences. His outstanding research publications are in prestigious journals, including the Lancet Neurology. Moving on to introducing Professor George Dias. Professor George Dias is a professor in clinical anatomy and biomaterials attached to the Department of Anatomy, School of Biomedical Sciences, University of Otago, New Zealand. He is also a member of the advisory board of KDU-CARE. Professor George Dias is a clinical anatomist with wide experience in oral and maxillofacial surgery. He has published extensively in clinical anatomy, paleopathology, and biomaterials. He has 91 publications, including 4 patents, 7,416 citations, and an H index of 29. His current focus is translational research related to degradable natural polymeric bone substitute and dental tissue regeneration materials, magnesium-based biodegradable orthopedics devices, bioactive hydroxyapatite, absorbable suture materials with wound healing properties, and extraction of premium protein from keratin. So while warmly welcoming both of you, sirs, to this IRC 2021, I now invite you to chair this session, and over to you, sirs.
Right, Dr. Samrakun, thank you very much for your kind words. I'm humbled by your introduction, and also welcome everybody. I can see there's a large participation, 48 participants. Welcome to you all and good afternoon. In Sri Lanka, it's good night from me. And it's 8:30 here. Yes, and also welcome to this, the plenary session, the session on basic and applied sciences of the 14th International Research Conference of Sir John Kotelawala Defence University. Again, I would like to thank the organizing committee for the honor of inviting me to be co-chaired in this session with Professor De Silva. And also, we have 5 world-renowned speakers at this session, and I'm really looking forward to listening to their presentations. Now, before we start with the speakers, few housekeeping announcements. Now, number one is each speaker is allocated 20 minutes for their presentations, and we'll be very thankful if you stick to the time so that we can go according to the schedule. And we will alert you at 19 minutes, that's 1 minute before 20 minutes up. There'll be an alert, a ring, a bell will ring, and as well as at 20 minutes, there'll be 3 rings alerting you to of the time. That's about the time allocation. And also, we will have a question and answer session, and that will be after all 5 speakers have delivered their presentation. So please hold your questions to the end. There'll be a session where we will, there's time for you to ask questions from all 5 speakers. And yeah, so that's how this session will run. And if Professor De Silva doesn't have anything to add, do you have anything to add to this? Or I can...
No, you can continue first.
Right. Now I take this opportunity to introduce our first speaker, Professor Toru Takeo. I hope I pronounced your name right. He's a professor at the Center for Animal Resources and Development at the Kumamoto University in Japan. And Professor Takeo, his fields of interest are in the development of reproductive medicine and assisted reproductive technology. And Professor Takeo obtained his PhD from the same university, Kumamoto University. He has established a very robust system of mouse sperm cryopreservation, as well as in vitro fertilization using this sperm. It's a very robust model. For this model, he received a number of awards, earned the Young Investigator Award of the Japanese Association of Laboratory Animal Sciences in 2012, and in the following year, received the award from the International Society for Transgenic Technologies. And apart from that model, recently I developed a new technique for superovulation in mice, where over 100 oocytes can be formed from a single female mouse. And there are a number of others achievements. He's a board member of the International Society for Transgenic Technologies, a member of the Asia Mouse Mutagenic and Resource Association, and a committee member of International Communication Committee. Dr. Takeo has a very impressive publication record. He has over 1,200 citations and an H-index of 19. So without any further ado, I call upon Professor Takeo to make his presentation. Professor Takeo, over to you.
Thank you very much, Professor Diaz, for introducing me. Hi, everyone. Nice to meet you. I'm Toru Takeo. Can I share my presentation slides? Can you see my slides? Okay. Okay, I'd like to start my presentation. In my talk, I'd like to talk about sharing system of mouse resources using reproductive technology. I've divided my talk into 3 parts. First, I'd like to introduce what is mouse research. And then I'd like to talk about development of reproductive technology. In the last part, I'll introduce you our activities to share our technologies. First, I'd like to explain our strategy to promote health innovations. Previously, researchers have to prepare suitable mouse models before their experiment. It was a critical limitation to start their research. To overcome this problem, we established Mouse Bank. Mouse Bank supports logistics of mouse research. This system is very simple. Mouse Bank archive mouse models and supplies the mice to researchers. This system helped to quickly start the experiment researcher. And also, researcher can quickly start their collaborations. Mouse Bank is my main work to establish the system. Why we focus on mice? Mice give us very, very new discoveries. Many researchers won Nobel Prize, used mouse to find new discoveries. And the mice also useful for educations and to find new drugs. Why are the mice useful? We share 98% of our genes with mice. So mouse gene is helped to understand human diseases. To understand our gene functions, we produced knockout mice or knocking mice. If we make knocking mice or knockout mice, we found any diseases, we will use the mice as disease model mice. I'll show you the example. If we test the 3 compounds A, B, and C using cancer model mice, compound C recovers the cancer disease. The compound C will be the drug candidate. So disease model mice is very, very important in drug development. There's an international project for knockout mouse project called COMP. Since 2006, scientists around the world have been working together to generate knockout mice for everything. The COMP project brought to International Mouse Phenotyping Consortium, IMPC. IMPC built the first truly comprehensive functional catalog of a mammalian genome. In the IMPC, we systematically examined the phenotype of 20,000 knockout mice, one for each gene. This is a project of international collaboration of mouse banks called International Mouse Strength Resource, IMSR. IMSR produced mutant mice, and IMSR published the data of the mice on their website. Any scientist can obtain the mice from mouse banks via IMSR. Now, IMSR collected 77,000 strains of genetically engineered mice. Please check the IMSR website. This slide shows a member of the IMSR. Center for Animal Resources and Development is our institute. CARD is a founding member of the IMSR. This is our institute. CARD is the first mouse bank in Japan and play a role in the Hub Research Center for Medical and Life Sciences using genetically modified mice. CARD in Kumamoto University is located in South Perth in Japan. There is a 5.8 thousand kilometer distance between Kumamoto to Sri Lanka. There is a 3.5 hours time difference. Now we are 5:46. We have 2 animal facilities. This is the inside of the animal facilities. Our animal facility has a capacity to breed 50,000 mice under specific pathogen-free with automatic water supply system and ventilated cage system. CARD is a one-stop shop of mouse research. We support the researchers. For example, we produce, analyze, preserve, and supply genetically engineered mice to researchers. In the card mouse bank, we archived cryopreserved embryos and sperm collected from 4.6 thousand strains of genetically engineered mice. This slide shows the benefit of mouse banks. Mouse banks support resource availability, reproducibility, sustainability, avoidance of redundancy, and animal welfare. The mouse bank is a social system for life science. I'll move on to the next topic. Reproductive technology is necessary for the mouse bank system. Reproductive technology is supposed to efficiently storage, produce, and transport genetically engineered mice. In the mouse bank, we use cryopreservation of sperm and embryos, superovulation, in vitro fertilization, embryo transfer, and cold storage of sperm and embryos. Until now, we developed various reproductive technology, and we published 90 papers on reproductive technologies. In our study, we found very useful compound to improve reproductive technology. I'll show you some examples. First, we improve the low fertility of cryopreserved sperm. It was a critical problem that the cryopreserved mouse sperm reduced fertility. To overcome this problem, we developed new sperm cryoprotectant. The cryoprotectant composed of raffinose, skim milk, and algorithm. It recovers motility. I will show you the frozen salt mouse sperm and oocytes. As you can see, the motility is very good after freezing and thawing. However, the sperm has not good fertility. To recover the fertility, we investigate the Sperm Activator. Fortunately, we found cyclodextrin has a strong activity to induce sperm capacitation. Cyclodextrin is a very unique compound. Cyclodextrin can bind lipid in their hydrophobic cavity. Especially methyl beta cyclodextrin has a strong ability to bind cholesterol. After the treatment of methyl beta cyclodextrin to frozen salt sperm, methyl beta cyclodextrin removes the cholesterol from sperm membrane. The removal of cholesterol is very important to activate sperm. So this is the fertilization rate of cryopreserved sperm. After the treatment of MBCD, the fertilization rate rises to 60%. Next, we investigate oocyte fertilizer. We focus on the structure of zona pellucida. Oocyte is surrounded by zona pellucida. Zona pellucida looks network structures. And this is a molecule structure of zona pellucida. Zona pellucida is composed of ZP1, ZP2, and ZP3 proteins. We thought if we can loosen the network, sperm can easily penetrate the zona pellucida. So we used a chemical scissor to cut the network. What is chemical scissors? we use the reduced glutathione. The treatment of glutathione to mouse oocyte, GSH broke the disulfide bonds in the peroxide, and also GSH induced expansion of the peroxide. You can see the structure within the peroxide is loosened by GSH. This is a fertilization rate of frozen cells oocyte after the treatment of GSH. The fertilization rate was over 90%. We combined 3 techniques of sperm preservation, sperm pre-incubation, and fertilization medium. We established CARD IVF protocol to obtain high fertilization rate in cryopreserved mouse sperm. I'll move on to the next topic. We increased number of ovulated oocytes. Before, we used the classic method of superovulation using gonadotropins, ECG and HCG. After the treatment of ECG and HCG, female mice ovulate 20 oocytes. When we use 20 oocytes, the 20 oocytes, for 90% of oocytes is fertilized and 50% of embryos develop to pups. So finally, we can obtain 9 pups from 1 oocyte donor. It was not efficient. To improve the efficiency, we developed a new superovulation technique using the inhibin anti-serum. What is inhibin? Inhibin is not new hormones. Inhibin is discovered in 1970s. There's 2 isoforms, and the function of inhibin is downregulate of FSH production and regulate the ovarian folliculogenesis. This slide shows a mechanism to induce ovulation using inhibin anti-serum. In general, follicular stimulating hormone is secreted from pituitary gland. In ovary, FSH stimulates follicular development. At the same time, inhibin is secreted from ovary, and the inhibin suppresses the production of FSH. This mechanism controls the number of ovulations. Using the inhibin anti-serum, the effect of inhibin is blocked, resulting in excess level of FSH is produced, and the FSH strongly promotes follicular development. This mechanism is induced ovulation by inhibin anti-serum. This graph shows the number of ovulated oocytes. After the inhibin anti-serum and ECG treatment, almost 50 oocytes were ovulated in each mice. The effect was higher than IAS or ECG alone. Interestingly, we found more than 100 oocytes were produced at 4 weeks old by IAS and ECG. Using the technique, we can produce 45 pups from 1 oocyte donor. The efficiency was increased 50 times higher efficiency was achieved. Next solution is a transport of fresh sperm and embryos at 4 degrees C. Shipment of live animals is getting difficult. This is the Nature papers. Now, FedEx and the UPS will not transport mammals for laboratory use. This is a huge problem. If they don't transport laboratory animals, researchers cannot get laboratory animals. To overcome this problem, we developed the transport techniques of cold storage embryos and sperm. First, I'll introduce the cold storage of mouse embryos. This is a step to produce mice using the cold storage toothed embryos. We prepare toothed embryos by IVF or sow the cryopreserved embryos. We can transport the embryos at 4 degrees C. At the receiving facility, they perform embryo transfer and they can quickly reanimate in their receiving facility. In this technique, storage medium is very important. We found M2 medium maintained and developmental ability of cold storage embryos. In addition, we found adding an acetylcysteine to M2 medium prolongs the storage time, and acetylcysteine prolongs the survival period of embryos at 4 degrees C for 4 days. You can see after 4 days storage, we transfer the embryo to the recipient. We can get many pups by embryo transfer. Next, I'd like to introduce the techniques of cold storage of mouse sperm. In this technique, we use coda epididymis. Coda epididymis is an organ to store matured sperm. We transport coda epididymis to receiving facility. They perform IVF and embryo transfer. They can quickly expand the colony using the sperm. I will show you the cold storage sperm for 7 days. After 7 days, the motility is greatly dropped. To recover the motility, we use cold protective agents, that is DMSO and quercetin. When treat the DMSO and the quercetin during cold storage, the sperm motility is maintained for 7 days. This slide shows the fertilization rate. DMSO and quercetin maintains the fertility of sperm at 4 degrees C for 10 days. Before, we use the cold transport techniques, only domestic transport system. However, we prolong the storage period. Now, we use the techniques to international transport of genetically modified mice. Let's move on to the last part, technology sharing. We share our technology by publications, online protocol, and workshops. Our publications, you can see our publications in our website, or ResearchGate, or my Twitter. So if you are interested in our research, please visit our website. And also, we published online manuals. You can find the online manual by Google. Also, we had workshops since 2000. Now, about 700 people are attended to our workshops. In the workshop, we provide a lecture, and then we show demonstrations, and we support training. We had a workshop in Japan. In 2011, we started international workshop. This is the first workshop in Singapore, and then we had a workshop in Taiwan. 2013, our workshop moved to the Madrid. In 2014, we had the workshop in Buffalo, New York. And we had a workshop in Korea. We had a workshop at Institut Pasteur in Paris. In 2017, we went to the Texas A&M Institute in Texas. And 2018, we had a workshop at Jackson Laboratory in May. 2019 and 2020, we couldn't have the workshops because of the corona pandemic. However, now we developed an online workshop system. This month, we had the first online workshop in our lab. If you have interest in the online workshop, please contact me. Now, cat-mouse reproductive technologies are available around the world. And we joined the community of ICM Mouse Mutagenesis Resource Association called AMRES. We are making a robust network in Asia. This month, we published the paper about AMRES in Mammalian Genome. If you have interest in our activity, please read our papers. This is my suggestion. If you are interested in the animal experiment, we recommend to establish a mouse bank of Sri Lanka. This system will promote science, and mouse banks support international collaborations. And also, CARD have international partners. Now we are looking for new partners in Asia. If you are interested in the partnership, please contact me. I'll summarize my thoughts. Mouse resources available via mouse bank. Development of reproductive technology ongoing at CARD. And technology sharing is performed via workshops and collaborate with international partners. I'd like to say thank you to our staff and our collaborators. Thank you for your attention.
Right, thank you, Professor Takeo. Perfect timing, up to the second almost. It is almost like your bullet train, you know, it's exact time, you know.
Thank you.
So thank you very much for a very interesting talk. And also thank you very much for your kind offer to set up a mouse bank in Sri Lanka, maybe at KDU. I highly recommend the authorities here, I think, go for it, because as you said, transporting animals are becoming extremely difficult, and this will be a fantastic opportunity. Yes, thank you. So please join me in thanking Professor Takeo for his excellent presentation.
Thank you very much. Thank you.
Right. We'll have a time for questions. You know, in fact, I also have one or two questions. We'll wait for the end. Right. So our next speaker is next speaker is Professor RMG Rajapakse. And okay, Dr. Rajapakse, can you please unshare, because I lost my document. Can you please?
Sure.
I'll put it to my other screen. Then it should be all right.
Sure.
Now we can share the screen.
Right.
Yeah. So Professor Rajapakse is the senior professor in chemistry at the University of Peradeniya in Sri Lanka, and he also had done postdoctoral research. He has been a research fellow in a number of universities: Imperial College London, University of Manchester, University of Bath, University of Central Lancashire. In fact, one of my colleagues is the professor there. Max Planck Institute in Germany, and he's the visiting professor at the universities of in USA, Texas, in Arlington and Mississippi. Also, he's a visiting professor at the Research Institute and Electronics, Shizuoka University, Japan. And also he's a consultant in nanotechnology to a number of Sri Lankan universities, the Rajarata, Velasa, Sabaragamuwa in Sri Lanka. And also he's a consultant to a number of commercial industrial sector, TJ Industries, ATG Limited, Pogala Graphite, Sarasavi, and Kodagen Industries. And his areas of research are many. I'll try to summarize. Is photoelectrochemistry, electronically conducting polymers, LCD devices, gas sensors, photodynamic therapy, and also graphite, to expanded graphite and graphene, very relevant to Sri Lanka, intelligent textiles and garments, organic fertilizer, and many, many more. He has over 10 national patents, as well as 2 international patents. He has supervised over 30 PhD students, and he has a citation index, citations of over 2,500, H-index of 28, a very proficient publication record. And without further ado, it's over to Professor Rajapakse to deliver your presentation.
Thank you very much for very kind introduction, Professor DS. And let me share my presentation with you. And I'm going to talk on a general topic on improving national economy of our country through commercializing our university research output. So let's just briefly look at top universities based on world ranking. And as you see, I mean, top 10 universities, the top 4 are in US and then UK, University of Oxford, then ETH Zurich. And then you have 2 more universities, Cambridge and Imperial College in UK, and then Chicago, US, and then UCL, UK. I have plotted these top universities based on different countries. There are out of 100 universities, 27 out of which 27 are in US, 18 in UK, and 7 in Australia, 6 in China, 5 in Japan, 5 in Hong Kong, and 4 in South Korea. Likewise, even like we have 1 in Malaysia, and there are sort of considerable numbers in the sort of Asian part of the world as well. And Sri Lankan universities, of course, I mean, according to world ranking of 2021, Peradeniya is ranked as 1896, 1896, and Colombo 2113. Likewise, I mean, so we are within like Peradeniya is within first 200, and then these universities are within first 300, and other 2 universities, especially these newly established Rabaragamu and Rajarata universities, are also within first 5,000 out of 300,000 universities that have been taken for ranking. So this is out of like even within first 5,000 is a remarkable value, although it's not in the first 100 or 1,000. But according to Times ranking, we are between 400 to 500 different ways of ranking. And I just look at this nature on the number of publications in very high index journals made from Sri Lanka during the period from 1st December 2019 to 30th November 2020, like within 1 year. And you can see that physical sciences publication, there are 16 highly indexed journals, and also in life sciences there are 6, including Nature and Nature Medicine also, and in earth and environmental sciences there are 4, including Nature. So we see that we are doing really well. We are doing in really good research, worth of publishing in highest index journals. Now let me see my let me talk on my research, focusing on to my research and development, and with commercializable products, some of which have already been commercialized. So I work on, as Professor DS introduced, in various aspects of nanotechnology in diverse areas of research, but I'm going to concentrate on very few things out of these things for this presentation. Some conversion of local minerals to value-added nanomaterials, and also textiles and gloves, and particularly we have made a very special face mask, antiviral face mask called Respiron Nano AV99. I'm going to talk on that. So I started to collaborate with many lead in Sri Lankan scientists, starting from Professor Keith Tennakoon, and in diverse areas, engineers, medical doctors, allied health scientists, veterinary scientists, in very different areas, in different departments of my faculty, science faculty, and even I produce these PhDs. Now I'm collaborating with these my own PhD holders, PhD students who are now occupying very high positions in universities and research institutes. And also I collaborate with many scientists, starting from my own supervisor, late Professor John Albright. I collaborate with many UK scientists, US scientists, including these two scientists in the National Renewable Energy Laboratory, NREL, and these key scientists in Japan and two key scientists in Norway. And we are in this mineral conversion project. I collaborate with many, and I produced several PhDs out of this project on different minerals. Quickly, if I go to the conventional local graphite, expanded graphite and graphene products, actually we use the rejected graphite on stone pieces, the graphite attached to stone pieces in the Bogala graphite mine. And we developed a solution when you put that stone with graphite attached to it, graphite particles delaminate and go into solution. And when you stir it naturally, expands, giving highly expanded graphite. Graphite structure is just like a carpet. Large number of thousands, millions of cards are stuck one on top of each other. And what you do is this space between consecutive cards you increase. So that is then you tell what is called expanded graphite. This particular expanded graphite that we have produced, it has a remarkable properties, like 1 gram of this graphite is capable of absorbing 120 grams of oil. So if you try to recover spilled oils, so then 1 gram is enough to recover 120 grams of spilled oil. So oil spills are very common in the naval roads where oil transportation takes place. And recently in Sri Lanka, around Sri Lanka also, there also is a huge problem of this is ship wreck, disaster. So there also we suggested to use this expanded graphite entrapped in polypropylene mass to recover. Just you can put the mat on the oil truck, and due to the favorable interactions, only the oil molecules, not water, go into the expanded graphite, and you can take the mat and then squeeze, recover the oil, and put the mat back to recover more oil. So we had 2 local patents for that. And also another important area is like our mineral sands are sold without any value addition. Together with my colleagues, Professor Chandan Udawatta and Professor Rohana Chandrajit, we developed a very low cost process to convert this ilmenite, which is iron titanate, to pure titanium dioxide and pure iron oxide nanoproducts with titanium dioxide phase specific. Whatever the phase you want, you can get it. Whatever the nanoparticle size, you can get it likewise. So this very phase specific nanomaterials. So we have produced these nanomaterials. So for the first time, usually this conversion of ilmenite to titanium dioxide requires 1000 degrees and conc sulfuric or conc hydrochloric acid in sulfate or chloride process. What we used was diamine hydrochloric acid at 170 degrees. And using rotating autoclave, using mechanical energy, we were able to break this very strong structure of ilmenite and produce titanium dioxide nanoparticles first, and then from the remaining we made this phase specific iron oxide nanoproducts. So we have 2 patents and 2 national patents, and we have applied for 1 world patent for that. And prototype is now, I mean, we have informed that to the relevant ministry of industries, and they are interested in it. Now prototype is being developed these days. Because of this pandemic situation, this development is not happening as the way expected, but is progressing. So it is one of those success stories where once you do your research and you develop a very useful technique, then you have to convey that message to the relevant authorities to get the attention for the development of, for the upscaling, and then to integrate that into your processes for which you can convert your materials into more highly value-added products for value addition to your country and to improve national economy. And also in the Nepal area in Sri Lanka, we have apatite, that is hydroxycalcium phosphate, as you know. So I supervise several PhD students there to convert this impure apatite into pure hydroxyapatite nano needles, just like those that are present in your bones. And then we formed a research group comprising of orthopedic surgeons, biologists, nanotechnologists, and chemists, and also computer scientists, design engineers, and production engineers to make custom-made procedures for orthopedic transplants. So I have no time to go into details, but you can see that, we use this apatite. Finally, you get this hydroxyapatite nanoparticles, and depending on conditions, we can get a spherical or needle type of nanoparticles. And then we develop techniques to coat that on like a stainless steel or titanium alloy, 90% titanium, 6% calcium, 4% vanadium alloy, that are medical grade metals that are used in making prostheses. And this video will explain everything. So I will play the video. This is the group. Do you hear sound? Can you hear sound?
Yes, we can hear.
Cancer patients have. taken and prosthesis transplanted. Right. So this video, actually the song is in Sinhalese, but I mean, if I just quickly translate, it is like, I mean, we made a group called Bioprosthesis Group, where we made custom-made prostheses, made them biocompatible by applying hydroxyapatite nanoparticles on their surfaces, and then got all these reports for their safety, the adhesion of skin integration and toxicity, corrosion studies, and then we got ethical clearance that took long time actually, and then we transplanted 2 such prostheses made, one in that girl and also one in that patient who had a massive cancer. And there are more than 300 children with brittle bone disease, and we developed what's called telescopic prosthesis. Of course, although this project have proven that, I mean, we can do all these things, this didn't go beyond that. So for the commercialization part, we need more things. So this is an example where such an important project just like terminated without commercializing. And then to commercialize, very important to find commercial partners, particularly the private sector industries. So that is where I started to collaborate with various industries. And then they wanted me to be a consultant. One of those is ATG ATG Ceylon Private Limited, where they produce industrial gauze. They own 5% of the world market for industrial gauze. So these are the gentlemen, those are the managing directors of the ATG, and R&D director, one of my students who's doing PhD, and 2 of my students, they are senior research officers. And they used to work as junior research officers. They are now in US doing PhD. And they are, of course, I mean, I made super hydrophobic, just like lotus leaf, water repellent, and stain resistant gloves, antimicrobial gloves, anti-static gloves. So with that gloves, you can operate cell phones because these are touchscreen gloves. And also these computers, etc., and chemical resistant gloves. Also, we work on effluent treatment and conversion of biological sludge, microbial sludge to organic fertilizer also. But here the conversion of these gloves into antimicrobial gloves is timely because this has a huge market these days due to this pandemic. And then Lanka Transportable Limited, they are the industry producing coating zinc in galvanization process for all the Sri Lankan transforms of the electricity board, and their sludge is a gold mine. It contains iron chloride and zinc chloride. So we separated them and made magnetite nanoparticles from iron fraction and zinc oxide nanoparticles, and then we studied their antimicrobial efficacies. They are highly antimicrobial. Actually made a paint using this pigment and donated to some of those hospitals in Sri Lanka where to the COVID-19 wards to sort of control the spreading of this disease. And also you can make nice bricks using these things by adding this sludge in different proportions, and we have studied their strengths, and they're comparable to other concrete slabs that you make for paving like highways, etc. So this project is continuing because both these projects are continuing because we have the necessary backing from the industry. And Bogle graphite also I collaborate to make expanded graphite and graphene products. So due to time limitations, I'm not going to go into details. And the Code 10 is a new industry in Sri Lanka where they make the latest technology sport car in Sri Lanka. And they got the first place in the 2018 this exhibition that was held in Green. And this is Dr. Harsha Singh. He gave this is Professor H.M. Mandara and Professor Premaratne, one of my teachers, retired professors, both of them chemistry and physics. And actually, I'm the leader, and they are supervising these students to make zinc bromine flow battery and supercapacitors, etc., and now lithium-ion batteries. So we are now successful in making zinc bromine 4 battery in the prototype 1 kilowatt hour one, and we are sort of upscaling to make 10 kilowatt hour zinc bromine 4 battery to store the energy generated from solar cells, and to use in sport cars also. And TJ Lanka Limited is a textile industry situated in Avissaville, Free Zone, and we collaborated with them for about 10 years actually. So we made super hydrophobic and stain resistant fabrics, antimicrobial fabrics, anti-pilling fabrics. Textile pilling is a huge problem when you're wearing, the pilling happens. So we made a special method to unique method to make this tackle pilling problem, and also anti-static fabrics. And based on this knowledge that we gained over 10 years, producing more than actually 4 PhDs and 3 MPhils, and we were able to design and develop a novel 3-ply face mask. So that is when this COVID-19 problem came to Sri Lanka in March 2020, we started, despite having this the curfew, I got a special permission to work on these things. And we made this very special 3-ply face mask. So this video shows you actually super hydrophobicity. When you throw mud doesn't stick and chemical doesn't stick. I think I don't have time to. So these things are quickly. But when you put on to super hydrophobicity, mud doesn't stick. Doesn't go to PRS. Goes to jersey material. This one, small ones are non-treated ones. The treated one is large. If you put any stain, it will attract these 2 pieces where non-treated ones. On the treated one, just like on the lotus leaf, you may have spheres with no interaction, so it doesn't go. If you try to push over, it don't go. And also chemical resistance, nickel chloride solution, nickel chloride doesn't go there. So finally, I'm going to talk on this Respiron Nano AV very special face mask that we have designed. This is the group, myself, consultant radiologist Dr. Chaminda Herath, who joined with us to work on it, and my students who developed this face mask and developed 4 very special instruments to characterize. Those days there were no instruments, no places to get this characterized in Sri Lanka, so we developed these instruments. So this video actually will tell you
Respiron AV99 is an environmental friendly, biodegradable, low cost, reusable and antimicrobial face mask with water and blood repellency and stain resistance. It also repels and destroys any aerosolized viruses or bacteria. In this multifunctional face mask, the target has been to reduce both the waste burden and the impact on the environment. As such, cotton, a commonly available, natural degradable fabric material, has been used in 3 layers, nanotechnologically modified individually, to have unique inherent properties. The outer layer cotton fabric is super hydrophobic to repel any aerosolized viruses and microbes, as well as blood or any other water-based stains, comparable to a lotus leaf shedding water. Any microbes penetrating the outer layer will reach the nanotechnologically modified middle layer with multimodal antimicrobial activity. Micro and nanoparticles are used to partially block the pores of the woven cotton fabric via chemical bonding to a level of 300 nanometers, resulting in nanometric range filtration. The adjustable ear loops ensure tight fitting to the face of the wearer. A unique mask design and a metal or plastic noseplate addresses the problem of air escape at the nasal bridge. The innermost layer is made up of 100% hydrophilic cotton to absorb carbon dioxide and moisture present in the exhaled air. This face mask can be reused even after 25 cycles of washing or disinfection, at least for a period of 1 month. The nanoparticles used in these masks are safe in a wide range of applications, including medical and cosmetic industry. High airflow tests and wash cycles have proven that the chemically bonded nanoparticles are stable and do not detach from the fabric surfaces. The mask has medical grade characteristics with high resistance to flammability, satisfactory
I think the time is catching up, really.
and
Oh
is
yeah, I'm going
highly
to stop now.
resistant
Thank you very much.
to blood.
Right, so that's all that I wanted to show you. Thank you very much.
So, okay, thank you, Prof Rajapakse. Thank you for a very interesting talk, and also very heartening to see all those new developments that you are doing and these new inventions in Sri Lanka. And also I'm very intrigued by all these orthopedic devices that you are manufacturing in Sri Lanka. Yes, I'm sure there'll be a lot of questions at the end regarding some of this work. So please join me in thanking Professor Rajapakse for an excellent presentation. Now over to Dr. Ranil Silva to introduce the next speaker.
Thank you very much, Professor Laz. I would like to now have three more speakers, and the first speaker is Dr. Tracey Bradshaw. She's the assistant professor at the School of Pharmacy, University of Nottingham, UK. Her specific research interests lie in discovery and development of novel anti-cancer therapeutics to treat intractable and drug-resistant tumors. Agents under investigation embrace novel chemical entities, bivalent drug products, Malaysian rainforest natural products, and nanomedicine. The later fields specifically include the research of apoferritin encapsulated experimental and clinical imaging agents to treat and diagnose malignant diseases. Dr. Bradshaw is a member of Drug Discovery Group, and she has co-authored 130 peer-reviewed publications. Over to you, Dr. Bradshaw.
Thank you very much. I'm going to share my screen. Can you see this slide? And can you hear me all right?
We can see and we can hear you well.
Yeah.
Thank you. Thank you very much. And thank you for your invitation. So I'm going to be talking about apoferritin encapsulation of gerantonin A for transferrin receptor targeting and enhanced cancer cell activity. On the left, you can see ferritin, which is coursing through all our veins. And if we take out the iron, then we're left with an apoferritin protein shell. And this is recognized and internalized by the transferrin receptor. And in the middle, you can see our natural product that we're working with, gerantonin A. I'm trying to advance my slides, but I'm having some sort of technical issue. Oh, there we go. So we all understand the importance of natural products in medicine. They're an invaluable source of therapeutic agents. They're evolutionary fine-tuned. They possess vast structural diversity and serve as privileged scaffolds in drug discovery. And in fact, between 1940 and 2012, about 50% of the drugs that were introduced into the clinic were either natural products or inspired by nature. And natural products have a dramatic impact in cancer. We can obtain natural products from plants, from marine invertebrates, bacteria, or fungi. I'm going to concentrate on plants for this speech. So you'll already be aware of alkaloids that have been isolated from Catharanthus roseus in Italy. And from this plant species, there is vincristin and vinblastin, which are used in the treatment of leukemias and lymphomas and other solid tumours. So here is the structure of vinblastin. And its mechanism of action is that it binds to beta-tubulin and inhibits tubulin polymerization. So it destabilizes or depolymerizes the microtubules, destroys the mitotic spindle. So it leaves the cells stranded in mitosis. Another important class of antimicrotubules isolated from plants are taxanes. And Taxol was first isolated from Taxus folia. But now docetaxel is a semi-synthetic agent which is synthesized from 10-deacetyl bacatin, and this is found in the needles of the European baccata, the European true Taxus baccata. So this doesn't necessitate destruction of the trees. Here you can see the complex structure of Taxol. The mechanism of action of the taxanes is that they stabilize the microtubules, so they inhibit again tubulin, but they promote polymerization of microtubules and stabilize them, so they cause a G2M cell cycle arrest. And the taxanes are extremely important drugs in the treatment of many solid tumors, not least breast cancer, ovarian, and lung carcinomas. So I'm going to be talking about gerantonines, and there are 5 gerantonines, gerantonines A to E. We've worked on gerantonine A and B, and you can see the structures here, the differences, the epoxide moiety here in gerantonine B. And the gerantonines are isolated from Tabani montana corymbosa, and they've been isolated by Kwan Hon Lim. So here's Kwan Hon Lim, who worked at the University of Nottingham in Malaysia. And here's my PhD student from a number of years ago, Vijay Raja, who worked on gerantonine A. And here is Mohamed Kazad, who worked on gerantonine. So they looked at the anti-cancer activity and tried to discern the mechanisms of actions of compounds. We first of all ascertained that there was broad spectrum anti-tumour activity, and the first cell lines that we looked at are listed here: breast, colon, lung and pancreatic cell lines. And we can see that for the gerantonines and the acetate derivatives, which were synthesized because of their enhanced stability, we often see sub-micromolar GI50 values. So here we can see a dose-dependent growth inhibition, and this is our T0 value, and we have calculated GI50 values. That's the concentration that inhibits the growth by 50%. So we can see growth inhibition and also inhibition of colony formation, dramatic inhibition of colony formation using the GI50 and twice GI50 values. When we performed cell cycle analyses, we saw that there was a profound G2M cell cycle arrest. 24 hours, most of the cells are locked into the G2M phase of the cell cycle. If we treat for longer, we can see that we're seeing populations emerging in the apoptotic region. And we can see that here for all of the cell lines, actually. So here are the controls, 24, 48 and 72 hours. And here at 24, 48 and 72 hours, we can see most of the events are trapped in the G2M portion of the cell cycle, telling us that we're probably working with compounds that are antimicrotubule agents. We looked at the polymerization of tubulin into microtubules, and for this we had a positive control, the taxanes. Remember, they promote polymerization, and a negative control, no-codazole, which, like vincristin, inhibits polymerization. And indeed, we can see that gerantonine A and gerantonine B are profoundly inhibiting polymerization of tubulin to microtubules. And together with Anne Straub at the University of Warwick, we looked at we tracked the plus end. So we seeded tubulin and looked at the growth of microtubules in the presence of enhancing concentrations of gerantonine. We found that actually the IC50 value, the concentration that inhibited growth or dynamicity by 50%, was in the same region as the GI50 value, telling us that this is an important mechanism of action. And here we can see the growth of tubulin into microtubules. So this is the control, and this is the GI50 value, significant inhibition of growth and of tubulin protein. And indeed, we saw, when we did confocal microscopy, we saw evidence of microtubule disruption. So here we have a control, here's a cell dividing, and the chromosomes are aligned on the microtubules, and in the presence of our positive control vincristine and gerantinines, we can see multipolar spindles. For an example, it's multipolar spindles, and we can also see evidence of apoptosis with the membrane cleaving. Crystal structure analysis told us actually gerantinine B was binding to tubulin, and it was binding to the colchicine binding site. So colchicine is a clinical agent. It does have potent anti-cancer activity, but it's actually used in treatment of gout. Like all natural products, we thought that it possesses multiple mechanisms of action, a rich and diverse pharmacology, and indeed we can see inhibition of polo-like kinase 1, PLK1 activity, and PLK1 is a master regulator of mitosis. It's an oncogene, it's an oncogenic kinase, and it's also a validated target for cancer therapy, not least in glioma. So we see inhibition of PLK1 activity and expression. And we also see that the gerantinine here, I'm just showing you gerantinine B, induce, like many natural products, reactive oxygen species. So this may also contribute to the ensuing apoptosis. Here we can see our viable cell population, but following treatment with gerantinine, we see cells appearing in the early apoptotic and the late apoptotic phase. So we're seeing here that the gerantinines induce apoptosis, and this is shown graphically below. The dark blue is late apoptosis and the light blue is early apoptosis. And these are a number of cell lines, our colorectal cancer cell line, lung, and pancreatic. And this is associated with caspase activity. As you know, caspases are the executioners of apoptosis. And we can also see other markers of apoptosis, not least depletion of the pro-survival gene MCL1 and cleavage of PARP and caspase. So in summary, activity of gerantinines inhibit potent anti-tumor activity. They induce profound G2M cell cycle arrest, inhibit tubulin polymerization binding to the colchicine binding site. They inhibit PLK1 activity, generate reactive oxygen species, all leads to programmed cell death by apoptosis. So there are multiple mechanisms of action, a rich and diverse pharmacology. And this may mean that there is potential for off-target effects and systemic toxicity. So we need a cancer-specific targeting and a cancer-specific drug delivery system. So here I want to introduce you to Luda and Neil, my colleagues, and we work on apoferritin. And actually, Dr. Karupu was a pioneer in this field with us, using apoferritin, which is the ferritin minus the iron. We've taken the atom of the iron out. And Charla developed an apoferritin-based drug delivery system for gefitinib. So we can see once the iron is out, we have an 8 nanometer core, which is ideal for drug delivery. I want to introduce you to my student Haneen, who has used apoferritin to encapsulate gerantinine A for transferrin receptor targeting and enhanced cancer selectivity. So apoferritin is biocompatible, it's non-immunogenic, it's species-specific, biodegradable, of uniform size, and it's stable and robust. And so Haneen encapsulated gerantinine A acetate into this apoferritin protein core. So it's of nano size, as I said. It's comprised of 24 polypeptide heavy and light chains. And these polypeptide chains are able to disassemble under acidic pH and reassemble. And this gives us a tool to encapsulate agents. It reassembles and self-assembles to form a hollow protein capsules, which is perforated by hydrophilic and hydrophobic pores. And these pores or channels allow the transfer of small ions, iron, of course, and small molecules. It is recognized and internalized by the transferrin receptor, which is enhanced, it's upregulated in cancer cells. So why is it enhanced in cancer cells? Well, cancer cells have a high metabolic rate, they have high iron demand, and they are often hypoxic. And so they upregulate mechanisms to increase drug delivery of oxygen and nutrients. And one of these mechanisms is by enhancing upregulating transferrin receptor. Another important feature of the tumor microenvironment is the EPR, the enhanced permeation and retention effect, which will allow, through this leaky tumor vasculature, it allows internalization of nano-sized medicine into the area of the tumor. And of course, the retention emerges from the reduced lymphatic drainage. So we have encapsulated gerantinine to enhance its solubility and bioavailability and increase circulation time. It will protect the drug from the body, from elimination or metabolism. It protects the body from the drug. Tumor targeting will allow for dose reduction, and also it is able to overcome resistance mechanisms. We've shown that with other molecules, it's able to overcome drug resistance mechanisms such as P-glycoprotein. So Hanin used the disassembly and reassembly method to encapsulate 120 molecules of gerantinine A. By reducing the pH down to 2, the apoferritin disassembled, and then by slowly increasing the pH to pH 4 and pH 7.4, she was able to reassemble the protein capsule and encapsulate 120. Characterization of the encapsulated gerantinine A included native gel. So this is a non-denaturing gel. So we can see here apoferritin itself. This is reformed apoferritin following the pH-mediated disassembly and reassembly. And then this is the reformed apoferritin, including gerantinine A. And if you look under B, you can see that the gerantinine A has been encapsulated. Another form of characterization is by dynamic light scattering and zeta sizing. And we can see here that the apoferritin, the reformed apoferritin and encapsulated gerantinine A all show a very similar size distribution and zeta potential, telling us that the protein capsule is showing us the integrity of the protein capsule, and also indicating that the apoferritin has encapsulated gerantinine A. It isn't merely attached to the surface. So now we wanted to look at transferrin receptor expression in a panel of breast cancer cell lines. So Hanin is particularly interested in breast cancer. There are two methods that we looked at. One was semi-quantitative Western blot, which looks at the expression of transferrin receptor in the whole cell lysates. This is not particularly sensitive. But there's quantitative, more highly sensitive flow cytometry, which is looking at the receptors on the cell surface. So we have a panel of breast cancer cell lines, but the MRC5 is our non-tumorigenic fibroblast cell line. And you can see that levels of transferrin receptor here are below levels that are able to be detected by Western blot. And in the cancer cells, we can see to varying degrees transferrin receptor is expressed. By flow cytometry, here is our normal, our negative control track, and here is following inclusion with the transferrin receptor antibody. And you can see in all of the cell lines, except for the MRC5, we can see a shift to the right, which is showing us that we have transferrin receptors, high levels of transferrin receptors expressed on the cell surface. And this expression, we can see here, so to move that, thank you. The expression here shows we can see selective uptake of apoferritin. So we've labeled the apoferritin with 5-carboxyfluorescein, and we can see following 24 hours, we can see selective uptake in the two breast cancer cell lines here compared to our fibroblast cell line. So SKBR3 is a HER2 positive expressive breast cancer cell line. MDA231 is a triple negative. So we're using cell lines here of different types. And So the apoferritin has been shown to deliver enhanced levels of gerantinine A into the cancer cells. So here we can see this is internal intracellular gerantinine A following treatment of cells with just naked gerantinine A in blue and apoferritin encapsulated gerantinine A in orange. So how does that translate to growth inhibition? So if we just look at these two cell lines here, we can see that this is gerantinine A acetate alone in purple in both of the cell lines. And we can see that following encapsulation, we can see more potent growth inhibition, and we can see growth inhibition at very low concentrations. The green lines are apoferritin alone, and this is indicating to us that this is a biocompatible nanocapsule. So here is just a summary of the GI50 values of all our cell lines that we used in this study. And you can see in pink, this is the gerantinine A acetate alone, and in blue, it's the apoferritin encapsulated gerantinine A. And you can see in all the breast cancer cell lines, SKBR3, 231, MDA 468, etc., we can see enhanced activity and much lower, significantly lower GI50 values following encapsulation. This is not the case with the MRC5s. Remember, there was very low or undetectable levels of transferrin receptor here, and we can see, although gerantinine A does exert growth inhibition, we do not see enhancement of that growth inhibitory effect following encapsulation. So these were MPT assays, and we saw similar results in the clonogenic assay. So here is control. This is gerantinine A alone, and this is apoferritin encapsulated gerantinine here. So these are the control untreated wells, and here the cells have received 0.2 micromolar gerantinine A naked and encapsulated, and we can see here significantly reduced colony formation following treatment of cells with the apoferritin encapsulated gerantinine A. We looked at a few, we looked at mechanisms of action, and I just wanted to show you here that we can see downregulation of whole PARP and emergence of cleaved PARP. We see downregulation of the survival gene MCL1, and we can see downregulation at lower concentrations following encapsulation with apoferritin. And also we can see downregulation or decreased PLK1 expression. So the apoferritin encapsulation of gerantinine A acetate is enhancing these pharmacodynamic endpoints. So in summary, I just wanted to say 120 molecules of gerantinine A were robustly encapsulated. We see cancer cell line selectivity via transferrin receptor enhanced delivery. Apoferritin alone was biocompatible in our in vitro systems. We saw enhanced gerantinine activity in cancer cell lines, and this was evidenced by growth inhibition and inhibition of colony formation. I didn't show this, but cell cycle perturbation, PLK inhibition, and also apoptosis as demonstrated by MCL1 downregulation of caspase cleavage. So I'd like to acknowledge all my students, Vijay, Mohammed, but particularly Haneen for this work, our collaborators, my co-academics. I would like to say a big thank you to Dr. Kapoor for inviting me, and to you at this John Kotalawala Defence University, and thank you all for your time for listening to my talk. Okay, I can stop sharing now.
Thank you very much, Dr. Bradshaw, for that absolutely interesting talk. And also Sri Lanka has a wide range of natural products, indigenous products. And Dr. Purupu is also now in KDU-CARE. So we are really, actually, Professor Charitha will be very much interested to see, a collaboration between your institute and KDU, because we also have so many plans to offer, and Anchala is with us now.
Thank you very much. It would be an honor. Thank you.
Thank you. So then we come to our next speaker. Our next speaker is Dr. Vijay Pal Singh, and he's the assistant professor in Academia of Scientific and Innovation Research and Veterinarian, CSIR, Institute of Genomics and Integrative Biology, New Delhi, India. His expertise area of research are laboratory animals, science, animal welfare, animal antimicrobial resistance, and food safety. He mainly involved with mice models of asthma, obesity, metabolic syndrome. He has conducted several international courses and laboratory animal sciences including the topic of experimental design, sample size, and statistician. He's an active member of many organizations. And over to you, Dr. Singh.
Thank you very much, Professor Ranil Vikram, for introducing me. Now, I hope you can see my screen.
Yeah, we can see.
Yeah.
Okay. Yeah, just a minute. So thank you very much again, Professor, and also for the invitation. Good evening from India, Delhi. So today I'm going to talk about my experience with the food and health when I worked with the Food Safety and Standard Authority of India, Government of India, and Ministry of Health and Family Welfare. What is the problem? There is a great acceleration in the global food system, and now we see that the carbon dioxide emission is increasing, ocean acidification is also rising, energy use is going high, tropical forest loss is also going high, and water and fertilizer usage is also going high. And we are not yet bending the curve on unhealthy and unsustainable food system. It is going up and up, as you have seen from my previous slides. So what is the challenge, and what is the scale of the challenge, and what is the problem? All over the world, you will see that there are 2 billion people which are lacking micronutrient like iron and vitamin A. 155 million children are stunted, 52 million children are wasted, and nearly 2 billion adults are overweight or obese, and 41 million children are overweight. 88% of countries face a serious burden of either of 2 or 3 of these forms of malnutrition. So you see, the problem is very, very multi-angle. There are different issues. Like one way you have the obesity, on the other side you have the people who don't even have to eat, or some there is a micronutrient deficiency. To overcome this and to achieve this, we need to have a planetary healthy diet for nearly 10 billion people by 2050. And when I say planetary health, it includes the health of the planet and also the health of individual. Now, this is the healthy diet which have been described by Lancet Commission as considered to be the planetary healthy diet, which is consisting of the whole grain, tuber and starchy vegetables, fruits, dairy fruit, protein source, added fats, and also added sugar. Keeping this healthy diet or this planetary healthy diet in mind, we have to describe and see what is the basically problem with all the region of the world. So if you see the plate which is just in front of you, half of the plate should have at least fruit and vegetable and also the green. Rest comes the whole grain and very minute of starchy dairy and animal source protein. But on other hand, when we see this is our kind of a reference of healthy planetary diet, what are we eating? Now, if you compare and see that presently in North America, the boundary which is we called as healthy boundary, if you see 100%, the percentage in the consumption of red meat is very, very high, 638%. Whereas the consumption of starchy vegetable is 170% high. Whereas the consumption of egg is 268, poultry 234, and also 145 of dairy food, whereas the consumption of the vegetable, fruit, legume, whole grain is very, very low. So this brings us the problem into the focus, at least into North America, and I will come to some other group of the country where the diet is unhealthy and also unsustainable. Coming to sub-Saharan Africa, the consumption of starchy vegetable is 729% high, whereas other healthy, which I showed in the planetary healthy plate. Vegetable, fruit, and legumes should be there, which are not up to the mark and not up to the healthy boundary. Coming to our area, that is South Asia, the healthy boundary is crossing only on one part, and that is the starchy vegetable, whereas the consumption of all reference planetary health diet like vegetables, fruits, legumes, and whole grains and nuts are really low. So keeping these problems in mind, what are we doing in India? I'm going to share some of my case study with you, and also the solution that is basically being done through the regulation as well as some other interventions like dietary shift. We have to shift our diet to the planetary healthy diet. I have shown you the problem where we are eating more and where are we eating less. Also, we should also avoid the wastage. Nowadays, at least 40% of food is not reaching to the stomach or not reaching the plate. It is being lost in the process as well as into the some other aspect of the food chain. Production is, again, we need to close the yield gap to the about 75% and balance sodium and also the phosphorus application. So these are basically the production system. I'm not going into detail, but we need to shift to the planetary healthy diet, avoid our wastage and increase the productions. What are the strategies? 5 strategies for a great food transformation. The first is to seek international and national commitment to shift toward healthy diet, healthy and sustainable diet, like nuts and also the vegetables and the fruits and the variety of food that we traditionally have in our country. But we are mostly now shifting to the more processed food or westernized food or food which is junk. Reorient agriculture priorities from producing high quantities of the food to producing healthy food. Rather than food which is quantity, we have to also focus on the quality and variety. Sustainably intensify food production to increase the high quality output. Like in our part of the country, in North India, we are producing either monocrop like rice or otherwise the wheat, rather than growing the variety or also different oil. And for that reason, now Government of India have to start various schemes like for increasing the oil production, as we are depending the oil mostly from the import from the outside. Strong and coordinated governance of land and ocean, as the biodiversity as well as the plant and animal life is going very monoculture and less. And this is also another example of the, at least we have to reduce our food wastage to half, in line with the UN Sustainable Development Goal, SDG. Now, as I told you, what are we doing in India to reduce and to reverse and to bend this curve and also make and promote the planetary healthy diet and also avoid the usage of the food which is not sustainable and not healthy. We started a program called as Eat Right India in attempt to change the conversation of food and nutrition, unveiling a new language of food and nutrition. Why do have we to do this? As I pointed out, the global data, the Indian data says that nearly 100 billion people are having this foodborne disease, and 6 out of 10 risk factor for burden of disease in India are related to food. So the risk is from the food which is related to the lifestyle disease as well as the foodborne illness are 6 out of the total. And we are wasting 40% of food, and 1 out of 2 children in population consume less than 50% of their daily need of iron, zinc, vitamin, folate and other vitamin B group. And overweight and obesity is doubled in last decade among the both men and women, and we are wasting food which is nearly 40%. So diet is deficient in macro and micronutrient triple burden, sometimes obese, sometimes deficiency, sometimes not utilization, also the disease. So managing all these things, we have inspiration from our father of the nation that have told, experiences taught me that it was wrong to have dwelt upon the relish of food. One should eat not in order to please the palate, but keep the body going. So it is important to understand that food is not only for taste. Food is to nourish. When it goes and bypass our palate or our tongue, thereafter it is nourish our body. Therefore, this purpose, we started a Swasth Bharat Yatra program to transform India food and nutrition environment through a judicious mix of regulatory, capacity building, collaborative and empowerment approach thereby protect and advance the public health and improve the quality of life across all walks of life. And tagline was Sahi Bhojan Behtar Jeevan. On English, we can say it is Eat Right India. Why we did this Eat Right India program? To reduce the foodborne illness and also reduce the macronutrient deficiency and reverse the NCDs and also reduce the carbon footprint, ensure more sustainable food sector. This is we have done through matching the supply and demand alignment and also through the trade by the healthy consumers and weak wealthy businessmen initiative symbols and acronyms and also the marketing of Eat Right India. And we converge this with other various partner and also the government of India various program like National Health Mission, Fit India program, Swachh Abhiyan, Poshan Abhiyan and Jal Shakti Abhiyan. So these are the various programs which are going for this one. How will we drive the benefit out of it? It will be who will drive the benefit out of it? Because the people, partnership and private. How will it function? Engage, excite and enable with the stakeholder. What will be like 3C, culture and campaigns and convergence. Convergence with government program, campaign like Eat India and also the cultural changes. This is the logo of Eat Right India, which says that eat cereal and pulses in quantity, which is educate, fruit and vegetable liberally, plenty of water, food and drink high in fat and sugar very sparingly. But now you can see that we are eating a lot of junk food and also milk and milk product also to be very consume plenty and meat, fish and also moderately. So with these things, we need to say that our program Eat Right India is also leading to the planetary healthy diet, which is leading to more food, which is more sustainable food safety as well as food security. How are we doing this? Eat safe. Eat safe means we are focusing on personal hygiene and also the surrounding hygiene, combating adulteration, eliminate toxic residue in food and prevent hazards of using cooking oil. How we are combating adulteration? Through the providing kit to the school, kit to the campus, and also not allowing the oil to be used again and again. When oil is used again and again, there is a formation of protopolar compound and trans fatty acid, which are very bad for our health. Eat right, balanced diet, eat timely and diet diversification, eat local and eat seasonal. Removing the industrial trans fat. Now by 2023, we will have the zero trans fat into our food, which is just 1 year before the WHO announcement of reducing the trans fat, which have direct consequence or cardiac problem. Reduce salt, sugar and saturated fat. We are eating a lot of salt and also the sugar, which is not good, and also the saturated fat, which is causing the obesity epidemic. And as I told you before, there is a lot of micronutrient deficiency, and for this purpose, we are starting with Eat Fortified Staples. And Prime Minister have launched a program where all the rice which will be supplied through government will be fortified with vitamin A. Eat sustainable, eat local and eat seasonal. Local and seasonal is very important. If you are taking food which is from 1,000 kilometers away, is not sustainable. No food wastage, reduce cycle plastic, and also conserve water in food processing and reduce chemical in the food chain. How are we doing this? These are our initiative like Clean Street Food Hub. The street food which is there outside, the Pathela wala which are selling are being also brought into the regulation. Bhog, blissful and healthy offering to the board. If you go to the temple, you get some prasadam from there. So there also. So we are covering nearly every aspect of the safety. Healthy means reduce high fat, sugar and salt and trans fat by 2023. Sustainable, as I told you, share food, save food, share joy. Instead of wasting food, which is nearly 40%, we should share food with the different platform that we have. And also RUCO. RUCO stands for repurpose of used cooking oil. If you are used cooking oil once for priming, you cannot use it and sell it. We are making biodiesel out of it. So these are the interesting programs. There are the capacity development, outreach like Eat Right Campus, Eat Right School, Eat Right train station, Eat Right bus station, and also the network of professional, like network of professional on food safety and nutrition. We have a system based on that, and we are bringing all the food safety related professional into one system. We have started with hygiene rating, where you can have star of the hygiene rating. Through this program, all these food safety businesses are getting audited. And we are also combating adulteration through mobile lab. So lab is going from one place to one place. It is also in a small truck. RUCO to prevent this total polar compound in the cooking oil, and this oil is being used for making also the biodiesel. Share food, save food, and share joy is basically to avoid the food wastage, and this has been taken care by Indian Food Sharing Alliance, which collect the food from these all parties and some places where it is not used and rechannel it to the places where it is being required. Eat healthy is through the food fortification. So we have notified standard for 5 fortified staples like wheat, rice, oil, milk, and salt. And also we have a very interesting program called as Aaj Se Thoda Kam. So Aaj Se Thoda Kam means from now onward, if I am eating this much of sugar, I will eat less. If I am eating this much of salt, I will less. I will slowly reduce sugar, salt, fat, and sugar and syrup. So this program is being run by this our famous cricketer as well as this stars from the Bollywood. So it is also very much appealing, and people are starting to make it a point that they don't eat too much, and also we are starting labeling on it. Trans fat is also, as I told you, we are reducing because this is directly related to the cardiac problem. So this is interesting program, Trans Fat Free India. So as I told you, the cluster approach, we are a developing country. So a lot of places are there which are not controlled. Like on the street, you have Thelawala or street food vendor. For that, we have a scheme called as Clean Street Food Hub, clean and fresh vegetable, and also the meat shop, mithai shop, and bhog. Bhog stand for bliss and healthy offering to the God, because a lot of temple, gurdwaras were offering a lot of food to the people, and at the prasadam, they are also brought into the regulation so that we can have more of the food safety, security, and the sustainability. Coming to the academic institute, like Eat Right School, Eat Right Station, and all these also being brought into the voluntary certification, that these places, most of the people go out for work and also read, and also the university where the food is not the healthy one, and there is a lot of junk food which is being promoted by companies and MNC. For that, we have Eat Right School, Eat Right Station, Eat Right Tea Estate, and also other schemes which are going, and this also we are adopting for hygiene rating and certification. So this is the backbone of Eat Right India program. We have rich content, we have a good outreach program, and we have a partnership with all the stakeholders. We are bringing all our the Bollywood actor and also cricketer who can make this program very interesting. We are converging with the government. Converging government means various schemes which are run by different departments. We are converging together, and also we are innovating in the sense that we are doing rapid assessment of food testing, which can be done through the various mobile food safety lab, as well as our moving small kit. So we have developed a small kit which is called as food safety magic box. In that one, you can do 100 of tests at your very much in your need, as well as in your grip, so that you don't have to travel to other place. And also develop small booklet, which is very good for the people to follow. We are developing a lot of rich technical content, the content which can be used like video library, poster series, Eat Right books. And as I told you, we have developed a book called as Detection of Adulterant through Rapid Test. These most of the tests you can do at the home without going to any other place, like colors, which are artificial colors, or some of the things which have been added into tea. These are the resources which are available free of cost from the download. Thank you very much for the opportunity, and it is my great pleasure, and thanks to the Dr. Gihani Javura for giving me the opportunity, and the KDU University. Thank you, sir.
Thank you very much, Dr. Singh. It was an excellent speech and very timely, even to Sri Lanka. So thank you very much, and we are looking forward. I hope KDU will be working very closely with you in the future events. Thank you very much. So we have our next speaker is Professor Steinbusch. He is a professor in cellular neuroscience at Maastricht University, Netherlands. He is appointed Knight in the Order of Netherlands. Venezuelan lines. His research is focused on neurodevelopment, influence towards depression and neurodegenerative diseases. He has pioneered double doctorates in Sri Lanka, and he is also the founding director of European Graduate School of Neuroscience, and he is the founding editor of Journal of Chemical Neuroanatomy. Professor Steinbusch has thus guided 105 PhD students, including students from Sri Lanka for double doctorates, and he has a current H index of 99, 475 publications, and 39,602 citations. Over to you, Professor Steinbusch.
Okay, thank you very much, Professor Da Silva. I'm going to try to see if I can put my presentation on the screen. I'm a little bit doubtful now. Let me see if I can use this. There he is. So, ladies and gentlemen, yes, one moment. It's coming. It's starting to open. Okay, as you can see, I have limited my slides because I like to do some talking with you. And moreover, the idea is also that I'm going to stimulate PhD students or master students who are watching this event for me this morning or for you this afternoon, and maybe would like to join Master University in our double degree program with Sri Lanka. So I'm not going to give you a lot of details. I'm not going to give you a lot of different experimental results, but more reflecting on what we have achieved in the last 7 to 8 years. And as mentioned before by Professor Da Silva, I'm the founding director of Neuro, and Neuro is a consortium of schools in the EU region, and I will explain this later. So today I'm going to speak about that we like to work on research, basically using neuroscience education towards innovation, translation, but also important valorization. And I've been in Sri Lanka 2 years ago, 3 years ago, in which I will show some pictures about this too, in which I learned how you can use natural products produced in Sri Lanka as a guidelines to use a better cognitive behavior, motor behavior, and anxiety behavior. And like I mentioned before here in my presentation, our graduate school is basically situated in Maastricht. As if you notice a little bit the geography of Europe, then you notice Europe is situated, Netherlands is situated in the heart of Europe, and Maastricht is situated very close to the Belgian border, which is situated here, which is about 2 kilometers. We are very close to the German border, which is just 25 kilometers, and we're very close to Luxembourg and to France. And of all these countries, we have relationship with it. But in addition to these 5 countries, we have a large number of colleagues, both in different countries, including Sri Lanka. So Sri Lanka is also a very good and reliable partner in our consortium. I'll just give you the names here, and at the end you see DDP, which means a double degree PhD program, which we have established. So these are the 5 countries in Europe, which we are collaborating with. But I also would like to give you the old name. Basically, I still haven't used the old name because I noticed that Professor Da Silva very recently has moved to another university, and I couldn't find the name so quickly yesterday evening that I decided to keep the old name. But as you can see, you are in the list of what we call 8 associated partners in which we are collaborating with. And these 8 collaborating partners are not only in Europe, but mostly in Korea, in Japan, In Bangalore in India, we had just before that Professor Se from India who was talking, and we have also partners in China and in Brazil. So all over the world are partnerships. And why is this important? We really would like to expand our program and also see more students from Sri Lanka joining the program. We were looking for funding also to bring our seniors and students towards Sri Lanka. Of course, this school is focusing on neuroscience, because neuroscience, as we see it, is the central part of all studies, and we like to use these as mechanistic studies related to mental health and neuroscience. And I give you some examples. If we specifically look to Sri Lanka, we were interested to look to natural projects. We have looked in the past to green tea. We have looked to curcumin. We have looked to other products which are produced there and which we know they have a direct effect on mental health issue. That's the basic part. At the moment, we're studying a lot on COVID-19 and mental health, COVID-19 and depression, COVID-19 and neuroimmunology. So there are a lot of scientific methods which we use that have high priority. And you could ask yourself, maybe, why should we work with Sri Lanka and why should Sri Lanka work with us? Now, one of the very nice achievements which has been established in Sri Lanka by Professor Da Silva is, of course, that he established a very good facility for collecting brain tissue. This brain tissue is then being further studied by basically genetic studies, whole genome analysis, which are very interesting for us because we really would like to compare and together with the companies which we are working with to see also genetic differences between Caucasian in Europe and the United States, Asian, African, and even Indian population. So this knowledge which we like to receive is important to look to what we call personalized treatment and personalized pharmacology. We like to do this to publications, show you some results later, but we also like to do it to education, so some students, and to valorization, to meeting to start up with companies. How we do this? What's our vision? We like to do relevant research, and we do that with PhD students because we feel that they are responsible, and we as seniors are responsible in the career and the career opportunities for young staff who will follow up us later in their career. So the drive which we have in Neuro, which we have in Sri Lanka, which we have with our partner, is that we have, of course, we induce mobility, so bring students from one country to the other country. We like to share knowledge and expertise. And then particularly, like we recently, and I'll show you some pictures of that, recently we started with a joint PhD program. And the first student from Sri Lanka who defended her thesis at Maastricht University, I will present in a moment. We go further because we also like now to start with a joint research master. So that basically in this COVID-19 pandemic, we have learned much more to work much more with virtual system. So this online master education has the potential, has the possibility that we can train students from all over the world, including, of course, also Sri Lanka. And why is then a double degree program interesting? Basically, the students who will join our system, we will have then also a European degree and a European certificate, will enable, if they want to go for a career in Europe, the market easily. Because our joint double degree PhD is recognized, is certificated by the European Commission. Okay, so the education, like I mentioned before, is both on master and PhD level. It's both fundamental for those of one. We have also psychology and cognition included, and we have clinical neuroscience as well. So people can really choose if they want to do pure fundamental, they want clinical, they want translational or psychology. And for all of them, we have research training available, which can be found on our website. And of course, the major focus is interesting. The major focus is the same for all over the world, also including Sri Lanka, also including our partners in China, in India as well, is of course the aging society, which is dealing with disease. However, at the moment, we have a very strong link also to work with depression, because we noticed that at the basis of all Alzheimer's disease, Parkinson's disease, schizophrenia, but not so much multiple sclerosis, is depression involved. So we know that basically neurodegeneration is a disease which starting from the brainstem, and from the brainstem, it spreads forwards into the forebrain and inducing either Alzheimer's or Parkinson's disease. So I have a little definition about this. Yes, of course, within Sri Lanka, and I focus a little bit on this, they like to study, and we like to study also structure. Structure has been forgotten, at least in Western Europe. We focus at the moment a lot on biochemistry, neurochemistry, and try to neglect a little bit more the structure. Basically, at the moment, at the big EU grants systems, and also in the United States, a new term is used, and that's called systems neurobiology, systems neuroscience, which basically integrates morphology at the brain level, but also at the peripheral level, includes behavior, includes cognitive functions into one big, a big component, like the different neurological disorders, which is different from psychiatry. The neurological disorders is much more working with prescription of personalized drugs, while psychiatry is much more involved in working with neuropsychological treatment. And at the moment, we are very much interested also in developmental disorders, like ADHD, but also autism. So this is an important issue which we really like to integrate. And the question sometimes is, what is the relation between neuroscience and neurobiology? Maybe not so obvious for you, but biology looks more at the biology, at the mechanisms behind the systems, while neuroscience is much more related to how you can affect the systems and how you can repair the systems. So much more related to translational neuroscience. And like I mentioned before, the new parts in neuroscience at the moment are evolutionary neuroscience, computational neuroscience, biomedical neuroscience, and biophysical neuroscience. These are some new areas which we are introducing as well. I use them over here. As you can see, we have used now a lot on developmental neuroscience, which looks how the nervous system develops. We know that there are two important moments in each life, your life, my life. And one moment is when you're going to die, that's inevitable. But the other important moment is when you're going to be born. So the time of birth presets your further brain development. If there was a small hypoxia just before birth, with stress-smitten mother, will develop basically depression and will develop early Alzheimer, early Parkinson, early epilepsy. We like to study molecular neuroscience because we like to look to individual molecules in the central nervous systems to work together with the pharmaceutical industry or with basal herbal components to see how they can change and modify the molecular systems. Very popular with our partners at the industry in Korea and in Germany. We do neuroengineering, or better we call it at the moment, neurorobotics. So neurorobotics is basically that we make new small component chips which we can implant into the brains of of people who are suffering from neurodegeneration and restore and improve neuronal circuitries. This is something which is getting more important. Then, of course, we have the imaging, which I think is a little bit in decline at the moment. It's over its major reach. At Maastricht, we have not only the 3.7, the 7.2, but recently also the 9.4 Tesla machines to really have very accurate diagnosis in the brains of various people. And of course, a very booming area is neuroinformatics, in which we really can, in which you really can look to genetic changes and look how increases in methylation, increases in histoacetylations is changing the profile of a person. And with using CRISP technology, we also try now to use brain information and change brain information in people who are suffering from certain diseases. Then, of course, we have the classical systems. We have the neurophysiology using electrodes. We have the social neuroscience, which we know, of course, is used in the faculties of psychology and sociology. And as I mentioned before, we have the system neuroscience, which integrates all. It follows the pathways of data flow within the CNS and tries to define the kind of processes. This is something which our students will learn and which we're going to adapt as well in our program. Well, before going to a last part of a more amusement part or refreshing part of my presentation to elaborate what we are doing, I also like to emphasize to you what kind of disciplines would you consider to be most relevant training. And we did a little in our school and in Europe, a little investigation and asked questions to a lot of participants. And we noticed that most of the respondents, yes, they mentioned that they like to work in cellular neuroscience. That's what we work to, or in molecular neuroscience. As you can see, imaging is still popular. The psychology part is high, and the systems. A little bit less relevant is what you see over here. So the genetic neuroscience is basically not yet known as much, as well as the neuroinformatics. So all over neuroscience, neuro, the combination with working with our colleagues in Sri Lanka, with our colleagues worldwide, is a challenge. It's a good combination, and I would like to mention to all students who are watching, all colleagues who are watching, and who like to participate, to contact me directly. Now, I collected some slides from the past, and basically we started here in 2013, as you can see over here, in which we first signed an agreement, a memorandum of understanding, as you can see it here, between Sri Lanka and our European partners. And this was a very nice collaboration in which I was talking here with Professor Da Silva and his team, and we were there. Very nice workshop, which was called How Competitive Our Research Grant Application. And this was a workshop also financed by the European Commission. Now, from that was a beginning, which we started, and I had to get used, and I really appreciated the way you opened the meeting, which was for me rather new. This was another workshop in 2015, in which a so-called IBRO, the International Brain Research Organization, together with the Asian, I think it was the, I'm not sure, the Asian Schools of Neuroscience, which was in Colombo. And we talked again for the first time about national products. And I must confess, I wasn't at the beginning rather sure, but then we talked about with the former minister of Sri Lanka, both Mr. Kamarj and the former Minister of Industry here in Sri Lanka. And we had a good combination in which we really could see here the both ministers and the Prime Minister and the former Minister of the Primary Industry and the former Minister of Health. We had a signing ceremony, and the signing ceremony, which I also gathered, I really appreciated. It is a very nice gesture from Professor Da Silva over here to sign a meeting and to start a meeting. And I talked about this with my colleagues also in Europe. And we said we missed something. like this, and this was a very good adventure. Then after this meeting, 2 years later, we had another meeting in 2017, the symposium on traditional and complementary medicine. And I met 2 more students. Mr. He will sit in the audience, and I'm pretty sure now that he has a big smile now. So we have Lakmal here in Mexico, and we have Mr. Weikom over here in Naladar. But we also have here, and that's why I focus here, that you can see we do have a lot of different students from different countries. And I also met our first PhD students who are going to graduate here, Printa Weikom. It was the time when we had a meeting together with some Chinese students, another student from Sri Lanka, and I think this student she was from India. And in this one, we had the possibility to really set up the systems. I was interested to proceed and follow this up. And in my last slide, you can see me here as in my official role in Maastricht University. I'm officially the pro-rector of Maastricht University. I have here signed the signature of the Maastricht, but it's not important. Most important is this part. And as you can see over here, this is the official PhD degree, which was recognized to Printa Weikom. Basically, she did a PhD together with Professor Da Silva in and defended at Maastricht University. She moved further towards Canada and is now pursuing a PhD degree in Canada. So this is a very good way that you can see also the students what can be achieved. There are possibilities. Professor Silva knows how to handle it, and we really like to follow it. So if you want to have more information, my email address is here. We work on molecular, we work on imaging, we work on neurodegeneration, and we like to really see local differences between different populations worldwide. And we really encourage students to join and to make a career. We are there to help you further in your career. Thank you very much. And I close this. Let me see where I can close it.
Thank you very much, Professor Steinbusch. It was a very nice talk, and how Sri Lankan, especially the masters and the PhD students, could look forward for having a very much closer collaboration with Professor Steinbusch. And also, I would like to inform that KDU-CARE under Professor Charita is now looking forward to start MSc and PhD in neuroscience. So your contribution in this area will be absolutely appreciated, Professor Steinbusch, as in the past, what you have done. Thank you very much for a beautiful speech. And now it's open. We have, say, 20 minutes for panel discussion and summing up by the chair. So we would like, there are around 57 participants. So we would like them to ask questions, make comments, and because we had a fantastic 5 internationally renowned speakers giving us in a wide range of topics. So therefore, we would like from the others to ask some questions or make comments, please. Thank you.
While waiting for questions, can I ask a question first from Professor, let me get there, sorry. Professor Takeo, it was very interesting. So how does one sign a material transfer agreement with your institution to get the embryos and sperms? Is there a procedure? What is the procedure?
Thank you for your question. So I couldn't hear you. So could you repeat your question? Sorry.
Okay, I'll repeat. Now, in your organization, that you mentioned that with the parties which sign material transfer agreements, MTA. Okay, that you can get the embryos and sperms sent under cryopreservation to, is it only within Japan, or do you do foreign for foreign collaborators?
Yes, we have experience to send our materials to the foreign countries, and it needs a MTA. So now we have the many different type of resources. So it depends the depositor decides the MTA conditions. So before you use our materials, you have to check the MTA for each resource. So if you are interested in our resource, you can contact the depositor and make a MTA, and then we will send our samples.
Right, thank you.
Any questions?
May I ask a question from Dr. Tracey? I'm Rajapakse, and I was really interested in your research. Actually, we have done targeted drug delivery research using inorganic nanocarriers, like magnesium oxide nanoparticles, calcium carbonate nanoparticles. We use calcium carbonate nanoparticles for cisplatin, and magnesium oxide nanoparticles for doxorubicin, and keratin extracted from hair, and titanium dioxide also nanoparticles for Taxol likewise. And we have published all these things. We have determined like over 90% drug encapsulation efficiency, and we studied the release kinetics also, because these nanoparticles, particularly calcium carbonate and magnesium oxide, dissolve in the slightly low pH at the cancer cells, like 6.6 to 6.8 like. Whereas when you use a normal healthy cells, its pH is neutral, so therefore they don't dissolve. So that is the way the pH triggered release that we have studied. Is there any way that we could like collaborate in that area? Because we have published further papers on these articles.
That would be lovely. Thank you. You may be interested in some of the work that some of my colleagues are doing. Cameron Alexander. He works with a lot of inorganic nanoparticles as well. I'm quite new to this field and really I'm more a cancer pharmacologist, a cancer biologist and drug discoverer, and I'm quite new to the apoferritin field, and I'm finding it very, very exciting, but I could certainly put you in contact with Cameron Alexander. In fact, if I might, has that answered your question okay?
Yeah. Well, I mean, I will contact that the person that you reflected. Right. So thank you very much for that. And also one question, like wind blasting, that was actually first discovered in a Sri Lankan plant called minimal. It is a apogenaceae with the Catharans genus, right? Wind blasting. So that was first found in 1970s when I was a student in our university, actually.
Yes, vincristine and vinblastin are also in the Tambanai, Montana, Corymbosa. I mean, the structure of vincristine is very similar to two molecules of durantanine. Some really exciting work that we did, because we used vincristine a lot as our positive control in the early days, and we generated cell lines that were resistant to vincristine and showed that they'd upregulated the P-glycoprotein. But these cells were still sensitive to durantanine, so the durantanines were able to overcome the multi-drug resistant mechanism, which is good news for cancer treatment, but I guess bad news for potential systemic toxicity.
Okay, thank you very much for answering this. Thank you.
I think there's a question from Amana Ahmed. Please ask a question.
Yes, I'd like to present my question to Dr. Tracey again. I am a pharmacy undergraduate. I'm still in my third year and yet to graduate with my bachelor's. And your study really intrigued me, because it was a little relatable to my field. And what I would like to know from you is that how far along the development process is this product, and how long would we have to wait till we find this in the market? And I would also like to know, what is it other than specific targeting of cancer cells? Is there anything that sets this apart from already existing medications in the market for breast cancers and other types of cancers? That is what I'd like to know. Thank you.
That's a good set of questions. So unfortunately, the development process can last many, many years. And our problem is always trying to find appropriate funding. And at the moment we haven't got any. We are in a related program. We are looking at the in vivo tolerability of apoferritin as a drug delivery system, and the preliminary data are very promising. So I will have to write a grant application to pursue the encapsulated gerantinine, and then it's getting companies that are interested or trying to persuade our university that a patent, although I don't like the patenting system particularly, but the drug development process, I think, in academia is very tough unless you have collaboration with industry or support from industry. What were your other questions? A marketplace. Okay, so yes, there are many treatments for breast cancer, but unfortunately patients do develop resistance very rapidly. So a recent treatment for targeted breast cancer, for breast cancers that have upregulated HER2 receptor, is Herceptin, but resistance does emerge. So we can use Affibodies. So Affibodies are 6 kilodaltons long, and they will recognize certain proteins, and we have an Affibody to HER2, to HER1, HER3 and HER4, and we can attach these Affibodies to the light chain or the heavy chain of apoferritin. And I believe, and Charla will talk a little more about this tomorrow, but we've shown in vitro that we can elicit selective anti-tumor activity and can overcome certain resistance mechanisms. So that's a way forward to target specific treatments more directly to specific cancers. Was there another part of the question? I'm sorry, I can't remember.
I think I got the answer. It was what is it that sets this particular product apart from the already existing medications?
One other thing then that sets it apart is that it's species specific and it's biodegradable and biocompatible. I mean, you can hopefully in the future we would be able to take ferritin from the patient themselves. It's a quite straightforward process to take out the iron, and then we believe it's a really promising drug delivery system, not only for drug delivery but for imaging as well, for imaging tumours. So I think that sets it apart as well.
Yes, thank you very much.
There are a number of other questions. I think we'll move on. There's a question by Lakmal Gonavela. Lakmal, you can ask a question.
Thank you very much, sir. Actually, I want to raise my question to Dr. Vijay Pal Singh regarding his interesting talk. Sir, my question is now most of the South Asian countries, we are moving towards a westernized diet with having many junk food. So now your program, Eat Right program, how practical will be, like with the poverty in these South Asian countries, how will it be practical to implement in these countries? What is your view on that thing?
Yeah, interesting question, Lakmal. This is really not easy, basically, Eat Right program. Just I would like to say some few statistics that usually ICMR, Indian Council of Medical Research, said that the salt should be taken not more than 5 grams. And now, as of now, we are taking more than 15 grams. Same is resulted for sugar and also for the visible and invisible fat. Hence, we have an epidemic of NCD, diabetes, obesity, and all. To reduce this one, we need to change the strategy by regulatory as well as capacity building, because those who are selling the junk food, MNCs, they are having a very high budget for the advertisement. I can cite that one company have the budget of $8 billion to push their food, which is not really good for our health. My PhD was on obesity. So if you are taking one Coca-Cola or all these food kind of beverages, you are taking complete 1 day sugar need of you, what you need for 1 day. And this is in the form of fructose, which is bypassing the phosphofructose kinase pathway. So how are we dealing with it? First of all, we are leading a Eat Right India program through the capacity building and also through, like, suppose I give one example, Eat Right Campus. In our institute, we are telling people what is good, what is not good, what is hygienic, what is not hygienic. And then we are certifying that institute as Eat Right Institute. School also Eat Right School. So one way with this, another way we are having a regulation which says on the front of the label packing that this label have, this food have this much spoon of sugar and also this much spoon of salt. And also in very near future, we are going to have a sign like no smoking. Smoking is bad for health. Same is going to come very soon for the sugar and all this food. So it's a multi-pronged approach. First, we are doing capacity building, informing the customer so they demand the good food, and also through the labeling and regulation. So we are dealing from the multi-pronged way because India itself is sitting on this diabetic capital. And as you know, any of these diabetes, obesity comes, so it brings a lot of other diseases also, comorbidities. And you have seen in the corona also that those who are diabetic, the COVID is very deadly for them. So it's a multi-pronged approach, Lokmal, from regulatory and other aspect also, as well as through the capacity building and also awareness, demand and supply. We are empowering the consumer to ask for the good food rather than asking for the tasty food. Taste is only up to the then it is not. It is important to understand that. And most of the time we go for test. We are not hungry. We eat because of the test. So we are trying various approach. I can show it. So this is the recent, I don't know whether you can see, Swasth Bharat Yatra. We were cycling throughout India for 3 months, started from different regions, reaching Delhi, telling every nook and corner of people what is right and what is not, because people really don't know what they are eating is really right or not. Thank you very much.
Thanks. I think there are a number of people with questions. Next, Yunus Imran, you can ask your question.
Yes, thank you for the opportunity, sir. So this question is for Sir Professor Dr. Harry Steinbusch. So really, thank you and very fascinating presentation to us. You have given such a big opportunity to Sri Lankans in the field of neuroscience, because neuroscience is a really rare field to obtain a PhD or master's among the developing world. So here you have given the opportunity to already one student in double PhD and 2 are ongoing. But here my question is there any way or opportunity for the research master students for double PhD with Netherlands or any other your own related institutions?
Thank you for your question, Yunus. You asked me if there's a possibility to do a double degree master program. Yes, we have. So basically, we have not yet signed an official agreement, but we have already agreements with other universities, and we could easily establish this double degree master program as well. We also have funding available, some funding available to bring you from Sri Lanka to do your master program, which takes 2 years in Maastricht. It's called officially the Holland program or the Tulip program. These are the 2 names which we have for this one. So if you are interested, contact me and I will see that you can have a master program. We have basically 3 different ones in neuroscience. One is on cognitive neuroscience, one is on genetic and pharmacological neuroscience, and the third one is on basically related to genetics and imaging in neuroscience. So these are the 3 different portions which you can have, and there are possibilities. I'm not sure. Can you tell me a little bit? What is your background? Are you master now?
Sir, I am working under Professor De Silva on Parkinson's and cognition. I have done my bachelor's and I'm planning to enroll my master's with Professor De Silva. So if there are any opportunity to get a double PhD with Professor De Silva and yourself, that will be one of the wonderful opportunities.
So basically, what I would suggest is then that you will continue to do your master in Sri Lanka, but you add to this a research elective with one of our Huron partners, and the research elective will be then take you 9 months and will bring you to a double degree.
Thank you.
Okay.
Professor Rajapakse, you have a question for Professor Steinbusch?
Yes, please. Thank you. I have a question from Professor Steinbusch as well. Actually, now this is a very trendy area now, using AC impedance spectroscopy to determine biological functions like cancer detection and neuronal activity in neuroscience, etc. Actually, I have more than 40 years of experience of using this AC impedance in various devices like solar cells and physical devices, whole cells, many other devices conducting polymers. But now that the field is open for the biological systems, I would like to contribute my knowledge to develop these areas to solve these equations to biological systems. If you could comment on the application of this simple technique like AC impedance in your systems to understand the behavior of neurons, that would be very useful.
I expected already your question because I listened to your presentation, and I think this area, combining this area, is very important. Now let me ask you what. Let me phrase you correctly. Why? The trendy area which we see basically in Europe, but also I could see from my colleagues in Japan. Basically, I work with 2 universities or 3 universities in Japan, which is Tohoku, Kyoto, and Fukuoka, is basically that we go to personalized medicines. And the major trend, which I also like to induce in our partnership with Sri Lanka, is to use inducible pluripotent stem cells from patients to use these cells for a personalized drug treatment. Because I speak not to the students, a lot of students, maybe and basically from our figures, even some of our students who are watching us today have depressive moods. They don't feel well. They think, oh, this is nothing. Why should I do this here? So depression, we know now, is currently the most relevant, what we then call mental health problem. It's about 40% of all the people. Mentioning this, we do not have a good treatment. Some people say, well, if you don't feel well, we have antidepressants, but keep in mind, antidepressants are not effective. Mostly, they are effective only for 30%. And we are using drugs who are 50 years old. So what do we need are, first, new drugs, new therapies, which can be tested on individual cells of a patient. So we're going to use what we call drugs who have been used in cancer therapy, we're going to use in neurodegeneration, and we're going to test them on cells from individual patients. So this is something which I really would be eager to maybe join your experiments too, and that we have a little discussion about that as well. I really would like to see this. Thank you.
That's great. Thank you very much.
There's a question from the audience. Please go ahead and ask a question from the audience, the live audience.
Yeah, can you hear me?
Yeah, we can hear you.
Yeah, I'm Dr. Peshala from Kottala Defense University. So I would like to direct my question to Professor Rajapakse. That, sir, I just want to ask you that you are aiming for several commercial products, local commercial products. But the thing is that as we know that we have plenty of sunlight in Sri Lanka, and we all know that you are doing research on solar panels. So I would like to know, is there any hope for us to have a local produced solar panel in future in order to use plenty of natural resources?
That's a good question. Actually, one of my students who is now a professor at NIFS, Professor Kumar, actually together with me, he developed this making solar panel technology in Shizuoka University. After getting his PhD from engineering, he himself and his professor established an industry called SPD technology, where he was the in charge of making the solar panels. So we have the expertise. We have several in Jaffna. Professor Ravirajan's group also has the very good expertise. We collaborate with Norway, Professor Ireland, and Professor Ravirajan's group in Jaffna got a huge research grant for collaboration between Norway, Jaffna, IFS, and Peradeniya, and also in Indian universities, Kumamoto Institute of Technology. And so we can actually produce solar cells. And the thing is that we need to have some sort of support from support for research from government side. So we have basic, we have very good funding for basic research. But for the application upscaling like these things, still lacking. So as you say, we have to think about these things because we are importing solar cells from China these days. In our rooftop also we have solar cells silicon ones imported from China. Although we are doing work on licensed solar cells, so it's a time that we thought about your question. Thank you.
Right, I think Dr. Bradshaw, you have got a question to. Have you got a question, Tracey? No.
Oh, yes, please. I'd like to ask a question of Professor Steinbusch, please. I enjoyed all the talks. They were really interesting. Thank you. But Professor Steinbusch, you said that hypoxia, was it just pre-birth, could lead to early onset Alzheimer's and Parkinson's. I just wondered if you could expand on this a little bit, please. It's fascinating.
Yes, most definitely I can expand it. And I decided I could make a full talk about this, but I decided not to keep this. Now, we work in our hospital a lot with children who are born too early. So they are suffering from prenatal stress. And we also notice that these children who have with prenatal stress grow normally, but they experience when they are about 12 years old, because we have a 20 years follow-up study done, early signs of depression. And basically that type of depression is completely neglected. And that we will tell to the children, come on, behave. It's not too bad in the world. Just follow it. It's not treated at all. Now, if these children, according to the bar theory, will get a second hit, a second major event when they are around 30 years old, like parents are dying or any kind of emotional feeling, that second hit will induce a very early start of what we call neurodegeneration. And there, for these children, which we also have followed over the past, and we had some, I'll give you some examples, we noticed that Alzheimer can already start when they are 45 or 50 years old. And where do we have these children also? These are also children who suffer from what we call PTSD, postnatal stress disorders. Yes, these are children where, when you go way back 80 years ago in the Netherlands, when we were occupied by the Germans and the Second World War, and we had to suffer from what we call the hunger winter. But myself, I did research in Africa, where there was fighting about 40 years ago between the Hutsis and the Tutsis, and a lot of children, and we noticed that in that situation with early stress, mothers who were suffering from early stress, the offspring of these mothers are suffering from what we call developmental diseases like autism, but also ADHD, and also again depression. Now we are following that one, and we also can see now in that situation the same problems appearing. We also saw it in China, because we did also studies with the big earthquake about 50 years or 40 years ago, I think, in the western part of China, where people were also suffering from these events, and we looked to pregnant women at the time of the earthquake, what was happening. So basically what I was mentioning is stress in late pregnancy has an effect on how your brain will develop. Yeah. So basically that's the important part, and you cannot treat it. So a healthy brain is important for a healthy life.
Yes, thank you. Thank you very much.
Okay. Right. Before we wind up, I think we have time for two more questions. Nalaka Vijayko, you can ask your question.
Yes, actually, my question is to Professor Harry Steinbusch again. Actually, sir, this is an addition to the question asked by Yunus Imran. Sir, like we know that there is a Euron certificate criteria for PhDs available in Euron. So I wonder whether there is a urine certificate criteria for MSCs available with urine.
You wonder if there's specific criteria. No, basically the criteria to get a European urine PhD certificate at Maastricht University is, there's basically very short, you need 3 papers. 3 first author papers with original data, no review papers, no ethics, 3 original papers. And we aim that the impact factor of these papers should have at least 4.0. So this is something, but some people get a little bit upset about this. How can we do this? Is this possible? Well, basically, we help you, we guide you, we train you. You're not alone in your PhD, you are with a team. Yes. And then with that PhD. Now, then you also mentioned, I think, if I'm correct, can you also have a PhD degree on an MD level? Yes. Is that what the question was?
No, actually, my question was for some people who will be interested in a urine certificate for their MSc, is there any ability for them to get, because for PhDs, yes, it's available in urine, that Maastricht University.
Oh, for the MSc, yes, for the MSc, I noticed that basic is almost the same question which I had before. We will also have a urine master certificate. And that master of PhD certificate enables you much easier to go for a PhD in Europe if you want, because it's recognized by the European Commission and it's basically it's registered over there, so that's easy to be done. If you want to do it an official degree, then you can do it, and I mentioned already before, there are 2 ways. You come for 2 years to Maastricht, or you stay in your university and you do 1 research elective of 9 months in our consortium. Yes. And don't worry about, I know some students think, yeah, how should I do this? How can I find funding? I always tell the students, funding is on the street. We always find funding for all of you. There's never been a problem to bring you over for 9 months. There are a lot of opportunities for the career of young students. But what you should learn, and I learned this from my colleagues, is you have to talk. You have to ask, you have to speak. If you don't talk, if you don't ask, there is nothing. So you have to learn, and that's a little bit a typically European way. You have to speak. You have to tell me, sir, I'd like to come, but I need money. Then we're going to help you. And if you said, sir, I would like to come, nothing, then nothing is happening. The action is to you. Yes. It's your life. It's not my life. It's your life. So you really have to take action, and we help you. We are there to help.
Right. I think even I would like to, the students who are listening, there are a lot of scholarships at Otago too, international scholarships for 3 years. So like Professor Steinbusch said, you should ask, you should apply, and then you can work through.
Yeah.
Right. There's one last question from the audience, live audience.
Actually, we have 2 questions, if you don't mind.
All right. Okay.
To Professor Vijay, actually, you said these cooking oil, which are reusing or used, can be used for biodiesel production or something like that. Because in Sri Lanka, we have this household habit of reusing the deep fried oil. So have you implemented any strategy to use this reused oil in India for production of anything else?
Yeah, actually for the used cooking oil, we have a program called as RUCO, reuse of used cooking oil. Means once you have any food business which is using 5 liter, more than 50 liter of oil per day, they have to keep a record. And also there is a machine which is, I'm forgetting the name, to check the total polar compound. So we have a regulation for the total polar compound should not be more than 25%. If their total TPC is more than 25%, you have to change. So for that, there is a biodiesel association. So these people who are using this oil, they can sell that oil to this biodiesel association and those who are collecting this. So we have made a system that those who are the big industry, they can store, and the biodiesel association people will come and collect, and they will make the biodiesel. So it is a, but they buy it a certain rate. I don't know the exact date, but there is a system in under RUCO. So they have to store this used cooking oil, and this will be collected by association, biodiesel association, and they will make the biofuel out of it. So there is a regulation system, and there is a collection point, and also they are paying for it. So there is a complete chain on this.
Yeah,
Thank you.
That is very interesting. I think that is something.
Yeah.
That
In fact, Professor Steinbusch has to leave or another. Okay. So there's one more question from the audience, and there's one more
question
from Amana. If you can keep it short, we have to wind up this session.
Yeah, there's one more question from audience. Yeah, please go.
Thank you, sir. This is Dr. Gihani Jayavira from Kotalawala Defense University. And thank you very much for all the resource persons for their interesting presentations. And my question is directed to Professor Rajapakse. Sir, your project on bioprosthetics aroused my interest very much. And you mentioned that it did not work to the like the commercial scale, but it looks like those are really benefiting to the people who are in need. Was there a particular reason why it is not extended to the commercial scale or become popular?
Yeah, that's a good question, Dr. Gihani. Yeah, well, I mean, if you want to make something commercial, convert your research output into a commercializable product, there are several other steps to follow. For example, I mean, just because universities make thousands of inventions every year, but not all go into commercial products. Like face masks went in because of the need. And those days, I mean, I developed this project during the time we had this intense war. During that time, of course, I saw the tragedy of broken bones of soldiers. So then the interest arose quite heavily, and we formed a group, and actually we made 2 custom-made prostheses and transplanted in needy persons. But to go beyond that scaling up, you need state support, for which actually those days we approached the 11 government ministries, and also I mean, they advise us to find a suitable commercial partner, particularly in the private sector. That is the step that we could not do, because although we have spoken to many commercial partners, they were not sort of willing to take a risk, because this involves huge amount of capital at the beginning. To have this, I mean, nowadays, of course, you can do 3D printing and things like that. It's much easier. But those days, I mean, we have to use these machines in the engineering faculty, like milling machines, lathe machines, etc. Computerized lathe, they are very expensive. So the commercial partners did not want to take a risk. But nowadays, I mean, we can rethink actually. So I was suggesting to my group to rethink about that, to use 3D printing technology, because what we do is we have CT scans, in MRI and all this data, then computer scientists have a program to convert those data into missing bone parts. And using this data, the design engineers design to 1 micron accuracy, and then production engineers produce in the university. So in the university, we have all this equipment and facilities. And also then they give these prostheses to us to make them biocompatible by depositing hydroxyapatite nanoparticles and polymethyl methacrate polymono, just to resemble hydroxyapatite nanoparticles and collagen polymer that you have in your bones. And then we give it to biological team to study toxicity, cell adhesion, what you call osteointegration, and then we do corrosion studies. And then, of course, I mean, we give everything after confirming all this is to the orthopedic surgeons. So they got actually ethical clearance. After getting ethical clearance for each and every one that we develop, then only they can transplant. So, so the process involves large number of steps. That is why, I mean, that could not go beyond that.
I think, yeah. Thanks. There's a last question. Keep it short. Aman Ahmed, yeah, and we have to wind up after that.
Yes, thank you so much for the opportunity, even with the limited time. My question is for Professor Takeo. I'm sorry if I pronounced the name wrong. And I was wondering about when you presented all the fascinating facts about the cryopreservation of the sperm of mouse and how it decreases the fertility when it's thawed. I was just wondering whether it also affects the human sperm, because cryopreservation is an option for cancer patients before they go into surgery. I have heard of that. So does it affect human sperms as well? Because I haven't really heard that side of the story up to now until I heard it today. And I want to know if the chemical activators and the activation methods that you mentioned work for humans as well. Yes, that is my question. Thank you for the chance again. Thank you.
Thank you.
That's
Thank
a
you for your
long question.
Okay. Oh, we didn't test our chemicals to humans, so we need more experiments. But basically, our system of cryopreservation in vitro fertilization is similar. So I think the compound will be effective. Thank you.
Right. I think we had to wind up the session. Please, we want to thank all the 5 speakers for a very interesting, and it generated so many questions. I'm sure we could be talking to you a long time if you carry on. But I'm sure that the speakers will be very happy to respond if you write to them with or whatever it is. And so therefore, Dr. Ranil Silva, shall we conclude this session? Do you want to say anything?
Yeah, I would like to thank all the speakers. It was absolutely interesting, and we could see how KDU could get involved with further collaboration. So we thank you, everybody, for these wonderful talks and for collaboration. Thank you very much.
Thank you.
Ladies and gentlemen, we have now come to the end of our plenary session. It is now time to appreciate all our plenary speakers and the session chairs for this lovely contribution towards the basic and applied sciences session of this conference. It was indeed a fruitful session. We will deliver all the token of appreciations to you soon as possible. We would also like to thank the track coordinators, Dr. Kanchana Bandara and Dr. Kalpa Samarakun, and also the committee members for their tireless efforts to make this event a success. The technical sessions of the Basic and Applied Sciences track will be held tomorrow, starting from 8:30 a.m. onwards, Sri Lankan time, and the Zoom links will be shared on all the social media platforms. We will now begin the poster session, so you may continue remaining with us. Once again, I would like to thank all of you for your participation physically as well as virtually. Have a good evening. Thank you.
Thank you.
Mali Jayasingharachi, who is a senior lecturer of KDU-CARE. We have next Dr. Peshala Kumari, who is a senior lecturer of the Faculty of Allied Health Sciences. And we also have Dr. Samira Senanayaka, who is a senior lecturer of the Faculty of Allied Health Sciences. The first paper ID would be number 34, with the title, An Analysis of Sports Psychological Skills of a Sample of Hockey Players in a Selected State University in Sri Lanka. You may start now. Is the presenter available? DRSW Disanayaka, you may share your slide now.
Copy skill inventory questionnaire used to gather gather data. Here I included psychological skills were divided into 7 subscales, each and every subscale and data included for questions. Coping with adversity, coachability, concentration, confidence and achievement motivation, goal setting and mental preparation, peak under the pressure, freedom from worry. Then welcome to my presentation. First, let me introduce myself. I am Shalini Disa. Welcome to my presentation. First, let me introduce myself. I am Shalini Disanayake from Sabaragamu University of Sri Lanka. My topic is an analysis of sports psychological skills of a sample of hockey players in a selected state university in Sri Lanka. First, introduction. Sports psychology. Sports psychology is the study of how psychology influences sport, athletic performance, exercise, and physical activity. Then next slide, you can see my research overall and specific objectives. Then methodology. In here, I included research design, study population, study sample, data gathering techniques, and data analysis technique. Procedure. ACSI questionnaire, it means athlete copying skill inventory. Questionnaire used to gather data. The psychological skills were divided into 7 subscales. Each and every subscale included 4 questions. Then the 7 psychological characters are coping with adversity, coachability, concentration, confidence and achievement motivation, goal setting and mental preparation, peak under the pressure, freedom from worry. Then results and discussion. First one, data analysis of ACSI score and hockey players playing experience. According to test results, p-value equals 0.018 and it is less than 0.05. It means there is a significant difference between hockey players playing experience and their psychological skills. The second one is ACI score and hockey players playing positions. According to the test results, p-value equals 0.871 and it is higher than 0.05. It means there is no significant difference between hockey players playing position and their psychological skills. And third one is ACI score and hockey players gender. According to the test results, p-value equals 0.000 and it is less than 0.05. It means there is a significant difference between hockey players psychological skills and players gender. Conclusion. There was a significant difference between psychological skills of a selected state university hockey players in Sri Lanka. Male hockey players have better psychological skills than the female players. According to the players' experience, there was a significant difference between psychological skills and players' experience. But there was no significant difference in psychological skills and players' playing position. There was enough evidence to reject null hypothesis and accept alternative hypothesis. And in here, I included some references that used in this research, and thank you.
Okay, now the paper is open for questions. I would like to ask you, can you please elaborate or explain more your conclusion? Because you have just given it as the results of this study confirms that there are differences in sports psychological skills in selected state university hockey players in Sri Lanka.
Yes.
Yeah, just explain it a little bit.
It means I get the 3 specific objectives on according to my topic. Then I analyze according to their playing position how the psychological skills depend on their psychological skills. Then according to their playing position, I analyze how they are depend on their psychological skills, and according to gender, how the difference between the psychological skills of the hockey players.
Okay, then the psychological skills, you have taken all these measurements together. Then you are giving that name psychological skills then, right?
Yes, yes.
Any more questions?
So, can you tell me why you chose to summarize everything and present as psychological skills instead of coming up with individual psychological parameters?
Sorry?
You summarized everything under psychological skills, and you have done your statistical analysis based on psychological skill as a whole. So can you tell me why you chose to make the analysis using everything as a whole instead of comparing individual psychological parameters?
I think the major psychological skills included in the 7 subscale. That's why I included all the things in one.
Right. Okay. Thank you.
Thank
you. Can we have an idea about the age group of the sample? Age groups of the participants?
In university level, it means age between 18 to 25.
Okay, thank you.
Thank you.
We will now move on to the second abstract. The test battery evaluate muscular strength and endurance of the abdominals and hip flexor muscles. This is paper number 38. You may now share your poster with us.
Right. Can you hear me?
Yes, we can.
Yeah, okay. I am Suraj Chandana. My research topic is the test battery evaluate muscular strength and endurance of abdominal and hip flexor muscles. In Sri Lanka, actually, we haven't our own test batteries to evaluate human performance. Especially for the youth people, we haven't any test battery. Therefore, however, we used foreign-based test batteries. According to the foreign-based test batteries, there are anthropometric measurements. That means the body segments, masses, and length, with compared to our Sri Lankan context, different. Therefore, we need a different test battery to evaluate the, especially for our youth people, muscular strength, endurance, and abdominal and hip flexor muscles. Then I use actually 405 males and 306 females from Sri Lanka, youth people. And all participants know known and apparent musculoskeletal injuries and able to perform the exercise correctly. The exercise protocol for sit-up is laid down on back, knee flexion about 90 degrees, and stabilize the lower body. Feet are touching entire period of performance. Keep hand behind ears without pulling neck. Lift head and shoulder blades from the ground and perform maximum repetition within 30 seconds. After that, this is the data presentation. The red color represent the male performance for the sit-up. This next one represents female's performance. So average male performance is 17 repetitions per 30 seconds and 13 for the female. This is the test battery, new test battery for the Sri Lankan people. So this is, as you can see here, 16 to 17 and 18, 19 maybe the average level of the performance. Female here 12 to 15 is average. Then after that I selected 68 students from University Sabaragamuwa. Then 68 students did Euro test battery, and we evaluate the relevant muscular strength endurance of abdominal and hip flexor muscles. Then 68 students qualified from Euro test battery. However, the new protocol, only 61 students were.
Thank you very much. Your time is up, right? Now,
Okay, thank you very much.
is open for questions.
Yes, I have a question for you. Now, when you said you're using a new test battery, a test battery involves a sequence of exercises, not just sit-up tests. So,
okay.
in your research, you have only used a sit-up test and you have called it as introducing a new test battery. Is this a valid statement?
Yeah, because I will explain it. Actually, this is we focus especially muscular strength and endurance, abdominal and hip flexor muscles. This is kind of quite the mixture in muscles. Therefore, especially for the test youth people's these particular strength and endurance, we need a kind of a test. We call test battery. We call test battery for that particular area.
And one more question. How now here you have done this for 28 students. So that is your sample size, right?
No. Sample size is 405 for male and 306 for female.
So can you generalize this results to the whole population then?
Because I selected more than 1,000 people in Sri Lanka. However, this according to this protocol, lot of people cannot do this particular exercise. Therefore, for the statistical analysis, I use only the correct performance and screen the all correct performance and evaluate it.
Okay, then thank you very much.
Yeah.
Okay, thank you. We will now move on to paper number 64, catalytic reduction of methylene blue by magnite silica composite. You may share your poster now.
I hope you can see my poster.
Yes, can you please enlarge it full screen?
Okay.
Presentation. Yeah, the presentation at the bottom. Yeah, he cannot see. No. Okay.
Okay.
We will share your presentation.
Can you see? Can you see the presentation?
Yes.
Okay. My name is Nadishan Vijay. I am from the University of Colombo Department of Chemistry. My research project is the catalytic degradation of methylene blue using iron oxide nanosilica composite. As an introduction, water is the most important natural resource in the world. Water that has been adversely affected in a quality by pollutant is named as wastewater. The organic dyes are the major water pollutants. These colored effluents affect the photosynthesis activity in the aquatic life through the hindrance of light penetration. The catalytic degradation of organic dyes by nanoparticles or the conventional method in the wastewater treatment is an effective and rapid technique. These are the methodology parts. This is a major three parts. The first one is a rice straw was washed with tap water and distilled water and ethanol. Then replaced 10% HCl and then washed with distilled water again neutralized pH value. Then carbonized at the 600 Celsius degrees to obtain silica. The prepared silica was replaced 3 sodium hydroxide solution to prepare the sodium silicate solution. To this solution, ferric solution was doped added and continuously stirring. The final suspension centrifuged and washed with distilled water under sonication. The final product contains two types of precipitate. The one type is a brown color precipitate and other type is a dark brown color precipitate. The second step is characterization. The synthesized nanoparticles characterized using scanning electron microscopy, XRD diffractometer, and FTIR spectrophotometer. Third part is a catalytic degradation of methylene blue using this synthesized nanocomposites. For this method, a reaction mixture containing 50 ml of 40 ppm methylene blue solution and 0.31 ml of sodium borohydrate containing solution, add catalyst 50 mg was added. Then 2 ml aliquot was withdrawn at the given time interval and determine its concentration using UV visible spectrophotometer. The influence of the methylene blue concentration, sodium borohydrate concentration, catalyst dosage, reaction temperature, pouring salt and ionic strength of the methylene blue degradation were studied under these conditions. These peak at the SCIR at the 543 confirmed the bond formation between silica and iron oxide. At the peak at the 35.6 at this XRD pattern confirmed the formation of magnetite. These are the scanning electron microscope images. The size range is 100 nanometer to 200 nanometer in this nanoparticle. At the methylene blue degradation rate was improved with the increase in the catalyst dosage, temperature, and the amount of sodium borate limit. Parts which is decreased gradually. However, the degradation rate decreased with increase in the initial methylene blue concentration. The cation with a lower hydrated ionic radii and higher valency sufficiently decrease the degradation rate. As a conclusion, the optimum condition of the methylene blue degradation is the 20 ppm concentration of methylene blue concentration and 1 gram per decimeter catalyst dosage, 6.25 millimole per decimeter of sodium borohydrate at the temperature of 50 Celsius. The highest removal rate methylene blue was found by 99.89 with the 20 ppm methylene blue
Thank you
solution.
very much. Your time is up. So the paper is open for questions. Yeah, can you please give some information about sol-gel method? That's how you have prepared.
Firstly, I take rice straw. There is a prepared synthesized part, rice straw, and it replaces with 10% HCl. And they again wash with 2 pH until pH get neutral and carbonize to obtain silica. Then the obtained silica again replace PM sodium hydroxide to prepare sodium silicate solution. To this solution, ferric solution prepared separately prepared ferric solution and doped added white styrene. And final suspension contains two types of precipitate. It's deposit separately dry and again characterized using this technique.
Okay, and also can you please elaborate your conclusion some more?
As a conclusion, we can conclude that the optimum condition of this catalyst on the methylene blue degradation, the optimum condition is a catalyst dosage 1 gram per decimeter and 6.25 concentration of millimole per decimeter concentration of sodium borohydrate and temperature 50 Celsius. At this condition, the highest degradation rate and efficiency given by this catalyst system.
Okay, thank you very much.
Thank you.
Thank you. We will now move on to paper number 173, Effect of Stocking Density During Brooding Period on Welfare Conditions of Broilers, a case study.
From Rajapakse University, Department of Agriculture. Topic of my study is effect of stocking density during brooding period.
Yeah, we will share your presentation. Just wait a moment. Huh? Recording. Okay, we will share your recording then.
Okay, thank you. Good morning to all. I am Anthony Veerabahu from Rajapakse University. Today I am going to present about my research study which I did. Topic of my research study is effect of stocking density during brooding period on welfare conditions of broilers. Animal welfare is a balance between the animal itself and its surrounding environment. Nowadays, society has a trend towards for good welfare conditions in animal production. So, assessment of broiler welfare is important. Objective of this study was to assess the effect of stocking density during brooding period on welfare conditions of broilers. Moving to the methodology, 780-day-old broiler chicks were randomly stocked at 4 stocking densities as T1 50, treatment 260, treatment 370, and treatment 480 chicks per square meter at brooding period. These stocking densities were reduced by half at the second week of the brooding period. Experimental design was complete randomized design with 3 replicates per each treatment. Welfare parameters were measured at end of the brooding period and end of the finisher period. Food pad dermatitis, tonic immobility duration, and plumage cleanliness was used as welfare parameters. A final score for each replicate was taken for food pad dermatitis and plumage cleanliness according to formulas which were included in Welfare Quality Protocol 2009. And also to measure tonic immobility duration, the individual bird was placed on its back on a laboratory table. Then the bird was restrained with one hand on its sternum for 45 seconds while holding the head and neck. Then the duration of tonic immobility reaction was recorded. If the bird returns to normal in less than 20 seconds, the procedure repeated. Move into the results and discussion. The first graph show that tonic immobility duration was increased with increasing stocking density. The second graph show that plumage cleanliness was decreased with increasing stocking density. And also food pad dermatitis was not reported in any treatment. Moving to the conclusion, tonic immobility duration was increased and plumage cleanliness was decreased with increasing stocking density of the brooding period. Thus, it can be concluded that the lowest stocking density enhanced the welfare conditions compared to T2, T3, and T4 treatment. Finally, I acknowledge to National Livestock Development Board to allowing to conduct my research in NLDB Mahaberiyathana farm. Thank you.
Now the paper is open for questions.
So you have half the stocking density after 7 days, so in the second week. What is the reason behind this?
Yes, this farm practice.
And is it going to affect your results?
Yes, brooding period is 14 days But second week of the brooding period, we changed the stocking density by half because of the that farm reduced the stocking density by half at the second week of the brooding period. So this concept apply for the other 3 treatments. Due to the growth of the chicks, they change the stocking density.
And is there any recommended stocking density at the moment which are practiced by farmers?
So recommendation is 50 chicks per square meter, but farmers are practice about 100 chicks per square meter.
Okay, then thank you.
Okay, thank you. We will now move on to paper number 392, Injury Prevalence and Biomechanical Movement Analysis among Fast Bowlers in Cricket, a review of prevalence and risk factors. You can now share your screen with us.
Okay.
Okay. Can you see me, please?
No, we can't see your screen.
Yeah.
Can you?
Hello?
Yeah, we are going to share your presentation then.
Okay. Sure.
Yes, it's fine now. No?
You can.
Yeah.
Okay.
Oh. Okay, we will share your screen.
Okay, thank you, Miss. Good day, everyone. I'm Kia. This is my review study on injury prevalence and biomechanical movement analysis among cricket fast bowlers, a review of prevalence and risk factors. This study was designed to have an understanding on past studies which have been conducted on the injuries among cricket bowlers and the risk factors causing them, and also on biomechanics and bowling action. In foreign context, it can be seen that many studies have been done on these areas. But in Sri Lankan context, a lesser concern has been put on these areas of study. As in every active sport, cricket players are also facing frequent injuries in different types and different levels. So bowlers, as a major role in the play, they are using their whole body, and we can see in every cricket team and every single match series, there are many frequent injured players in the ground. So this study separately observed empirical data and methodological data regarding the studies considered, and it can be seen that most of the research studies have revealed the facts regarding the injuries related with lower trunk of fast bowlers. And workload of the bowlers is also considered when assessing the risk factors of the injuries, providing considerably high and low workload as factors that increase the injury risk of the bowlers. And overuse is also considered as a risk factor according to every study. And in most studies,
the frequency of the injury occurrence is considered and assessed to make a supportive fact with comparing the workload and the movement patterns which are observed by bowling actions using various kinematics. And in most studies, video analysis has been used by the researchers for movement analysis. By observing these studies, it can be seen that fast bowlers are considered as highly vulnerable for injuries, but it can be recommended that spin bowlers should also be taken into consideration to assess the injury patterns among them also. And in Sri Lankan sport field should be more active in these areas of study, and it will help to reduce the injury occurrence that may affect the players' general health and also the sport performance of the whole country. Thank you.
The paper is open for questions now.
Can you hear me?
Yes.
So what was the underlying reason for you to do this review study, and what is the objective? Why did you do it? What is the outcome you expect out of this study?
Yes, sir. So in Sri Lankan cricket team, we can see more frequently, they are facing injuries and downfall of the performance. In the underlying reason behind their injury occurrence. So biomechanical analysis is, I think, it's very much necessary for in a situation like this, because treating only superficially is not enough to cure the reason, the cause completely. So I need to make a step forward to study regarding this biomechanical analysis, and I want to suggest some more successful remedies to stop the injury occurrence among Sri Lankan cricket team players.
Okay, so what would you suggest after completing this study, after completing the review, and after obtaining knowledge on what the foreign countries do? What are your suggestions for Sri Lankan cricket in order to improve their injury prevalence?
Yeah, I'm an MPhil student, sir. So I'm conducting my MPhil study regarding the injuries among other bowlers in Sri Lankan cricket team. So I took this review. I studied those past studies to take an understanding how to improve the performance in Sri Lankan cricket team using those factors.
Thank you. Now move on to paper ID 436. Comparison of insect assemblages on two invasive alien plant species, Annona glabra and Lantana camara, in selected habitats of the wet zone.
Can you see my screen?
Not yet.
Okay. Can you share the presentation on behalf of me, because there's trouble with my computer?
Yes, we can do that. Yes.
This one actually has This a video. presentation is based on the research project Comparison of Insect Assemblage on Two Invasive Alien Plant Species, Annona glabra and Lantana camara, in selected habitats of the wet zone. These two plant species were selected based on their distribution and abundance in the wet zone, and as they are the prominent invasive species which can be identified in the wet zone. For this project, we selected to study insect assemblage rather than insect community, because due to insufficient time, the interaction between invasive plant species and insects, as well as between insects that were found to occur on the plants, could not be investigated in sufficient details to call as community. The main objective of this project was to study insect assemblage associated with Arna glabra and Lantana camera in selected locations in the wet zone. Sampling sites were selected based on the preliminary field visits, and the selected sampling sites were locations in the Bellamvilla Athidiya Wetland Sanctuary in Colombo District and in Garanduwa Wetland Area in Matara District. In each site, selected plants were divided into 3 spatial levels as bottom, middle, and canopy layers, and observed for 7 months from May to November 2016. Standard collection methods were used and the preserved insects were identified in the laboratory. A binomial logistic regression model was used to find out the probability of occurrence of recorded insects in different orders being present in the 3 layers of each plant type in more and less disturbed areas. The results of the study reveals that insect assemblage of Arna glabra consisted of 10 orders of insects, while 8 orders of insects associated with Lantana camera at both locations. Also, family Chrysomelidae of order Coleoptera showed the highest diversity of Arna glabra, while family Nymphalidae of order Lepidoptera showed the highest diversity of Lantana camera plants. Also, based on the observation, some of common type of behaviors of insects associated with these 2 plant species were identified and graphed. Results of the statistical analysis that show that the most insect orders prefer to bottom and middle layers of the plant species in less disturbed areas, while only order Lepidoptera and order Hymenoptera prefer the canopy layers in both plants, and order Diptera prefer plants in more disturbed areas. Despite being invasive plant species, a high diversity of insects associated with Arna glabra and Lantana camera.
Okay, the paper is open for questions.
Can they become a carrier of those pests actually? These 2 plants?
Sorry?
Can they become a carriers for this pest actually? Because
Carrier of pest. Yes, they can be, but as I mentioned, because of the insufficient details about the native or the endemic and the pest species identification in Sri Lanka, we cannot say what are the specific species that can be acted as a pest based on these 2 plant species.
But you have given here, no? That most of them are pests, no?
So most of them are herbivores. So in Sri Lanka, the identified insect species are most of pest and endemic species. But in here, these associated insects for these 2 plant species, we can't say most of them are pests.
And is there any specific reason that you have selected these 2 alien plant species?
Yes, because these 2 plant species have categorized as one of major invasive plant species in Sri Lanka, and also these 2 plant species have highly abundant in the wet zone in Sri Lanka. But there are some studies about these plant species and in plant species, but there are very few studies on insect assemblage and these plant species. That's why I chose this topic.
Thank you.
Thank you.
Next, we will move on to paper ID 475. Determine reference intervals for selected clinical chemistry parameters using selected healthy adult population in Sri Lanka.
Can I start?
Yes.
Good evening, everyone. I'm Shanti Panaguda from Horizon Campus. My research topic is to determine reference intervals of selected biochemical investigation of selected healthy adult population in Sri Lanka. The reference intervals is crucial for disease screening, diagnosis, monitoring, progression, and treatment efficacy. Due to lack of locally derived reference values for the parameters, clinicians use reference intervals derived from Western population. Different studies also indicate considerable variation in clinical chemistry reference intervals by several variables in such as age and sex. This study was aimed to determine reference intervals values of selected clinical chemistry parameters of full blood count, ESR, TSH, full blood blood sugars, liver function test, lipid profile test, and renal profile test using selected healthy adult population of Colombo City, Sri Lanka. This may be the first private sector study to establish reference intervals using Sri Lankan population considering age and gender. Data were collected from existing database of Navaloka Hospital Laboratory for 8 months period starting from March 2019 to October 2019. Based on inclusion and exclusion criteria, 1,054 individuals records were ultimately selected in this study. Inclusion criteria included male and female elders between 18 to 80 years old and age less than 18 years, adults with common intestinal parasitic infection, hemoparasite, HIV, HCV, HBV positive, HCG positive for females, hospitalized persons, chronic diseases, cardiovascular diseases, kidney diseases were excluded from this study. All statistical calculations were performed on the Minitab version 17.3.1 software. Descriptive statistics was used to determine mean, median, 95% coefficient interval, minimum, maximum, and 2.5, 97.5% range of each parameters. P-value less than 0.05 considered as statistically significant. Going into the results part, there was significant difference between gender in the levels of eosinophil, basophil, hemoglobin, PCV, MCH, MCHC, RBC, MCH, MCV, platelet count, specific gravity, TSH, ESR, albumin, globulin, bilirubin, ALT, GGT, and GGT, AST, triglyceride, HDL, total HDL ratio, VLDL, urea, keratin, uric acid, calcium, and phosphorus. And there were also significant difference according to the age group, WBC, hemoglobin, PCV, RBC, MCH, MCV, platelet count, TSH, ESR, ALT, total cholesterol, triglyceride, HDL, LDL, total HDL ratio, VLDL, chloride, pH, keratin, phosphorus, FBS, and glucose.
Thank you very much. Your time is up. So your paper is open for questions. What are the criteria that you have used to select healthy volunteers, healthy adults? Which records you have used?
Only age between 18 to 80 years old and only healthy adults.
So can you call them as healthy adults?
Yeah.
Without any medical test or any clinical tests?
They are only test they only test
yeah, I have the same
selected
question
from the existing database.
Any questionnaire you have introduced them to, or the patients involved in this study, or records
on records?
Yeah, it is only records, but my concern is that you have selected
healthy adults from a Navaloka Hospital Laboratory database. So how come you have selected healthy adults from a hospital database?
No, you need to clarify inclusion criteria. You need to
Yeah.
define properly what's the base. We have basic actually. Why? How you have selected healthy adults? No, how you call them healthy adults?
What are the criteria?
Yes, you need to.
Yes,
You
healthy.
should put in inclusion criteria actually.
These persons only check I collected data from only full body checkup persons, not the hospitalized persons.
Okay. Okay. Are there any records beyond the reference limits? Any medical test?
No.
All the test records are within the reference limits results for all the individuals.
Sorry, I didn't hear.
All the results of the medical test that you have considered are within the reference levels.
These parameters are significantly different according to the gender and according to the age. I categorize it into two groups.
I mean within this reference level, that means healthy. They all are healthy people. After analyzing these records, you realize that they are healthy people.
Yeah, some are have some issues.
So you can't call them healthy.
Okay, then thank you very much.
Thank you. We will now move on to paper ID 530, comparative study on antioxidant activity of fruit peels and seeds of pomegranate cultivars grown in Sri Lanka. You can now share your slide. Are you there, T.I. Punchi Patabandi? Okay, we will share your slide.
Comparative study on antioxidant activity of fruit peels and seeds of pomegranate cultivars grown in Sri Lanka. Pomegranate is a rich antioxidant source with antiviral, antifungal, antibacterial, anti-cancer and anti-inflammatory activity. Since antioxidant properties of fruit juice and fruit peels of other varieties are known except for Culpitia hybrid variety, in this study we evaluate antioxidant potential of hybrid juice, peel and seed. Also we compare antioxidant potential between peel and seed samples of pomegranate cultivars grown in Sri Lanka. My sample collecting center was Culpitia Agriculture Research Station. Fruits from Culpitia hybrid, Daya and Nimali varieties were used. Methylate extraction was used to extract phytochemicals present in air-dried samples of peel and seeds. In order to evaluate antioxidant potential, I have conducted these 6 bioassays followed by a statistical analysis. When it comes to results, except for IC50 values, column height directly proportional to the antioxidant activity. Therefore, greater the column height, greater the antioxidant potential. So, pink color columns corresponding to peel samples are greater than purple color columns corresponding to seed sample for total phenolic content, for total flavonoid content, for ferric reducing antioxidant power, for proanthocyanin content, and also for total anthocyanin content. When it comes to IC50 values, since they are inversely proportional to radical scavenging activity, least column height represent highest radical scavenging activity. Therefore, pale sample process greater radical scavenging activity compared to their seed samples. So we can conclude that pomegranate peel process significant high antioxidant potential compared to seed for all three cultivars. When it comes to hybrid variety, column height corresponding to hybrid juice is less than both peel and seed sample for total phenolic content, for total flavonoid content, for ferric reducing antioxidant power, and also for proanthocyanin content. But when it comes to IC50 value, radical scavenging activity of hybrid juice is greater than hybrid seed, but still less than hybrid peel. To total anthocyanin content of hybrid juice, it is greater than both hybrid peel and seed. Therefore, we can conclude that hybrid peel possess significant high antioxidant potential compared to juice, but except total anthocyanin content.
The paper is open for questions.
So after coming up with your results, what would you recommend? What are your recommendations based on your findings?
Since we can observe higher antioxidant activity for pomegranate peel samples, that means usually it's a waste from the juice processing industry, discard. So since we are having more antioxidant potential compared to juice in this peel sample, so we can use them to develop some healthy products like antioxidant rich things, because antioxidants mean crucial things for our health now. So we can develop something like that using this valuable discard.
So when you say you use the peel, do you exclude the outside and take the inner component of the peel, or do you take everything together?
Hello, I didn't hear the question.
So when you say that the peel has a lot of antioxidant effects, do you take the peel as a whole, or do you extract the insides of the peel?
Like the thing I did was I air dried and powdered it. Then after I extract that powder to methanol.
So you didn't throw away any part of the peel. You took the entire peel.
Yeah, I took entire peel.
Yeah, actually, there's no statistical method you have mentioned here, and even p-values you have not mentioned. Without that, you can't use significantly the word actually, and how your extraction method influence on the maximum yield of this.
In the methodology part, I have mentioned that I used the Minitab to analyze my results, and my p-value was 0.8. I have mentioned it here, the p-value in the methodology part.
In the abstract, you have not mentioned, no?
Okay, I think I have included it to this poster, but I think I haven't included it as abstract.
And actually, Minitab is the software, not the
statistical method you need to specify.
Yeah, I did two-way ANOVA. I did a two-way ANOVA. Sorry.
Okay, then thank you very much.
Thank you.
Thank you. We will now move on to paper number 572. Variation of selected agronomic characters of traditional rice variety Masuran in response to different planting dates within a year.
Can you see my slide?
Yes.
I am Pushpa Kumari, attached Board of Study in Agriculture, University of Rohana. My research title is Variation of Selected Agronomic Characters of Traditional Rice Variety Masuran in response to different planting dates within a year. So I'll give brief introduction about this study first. We all know rice is one of the most important cereal crops and provide the staple food half of the world population. In Sri Lanka, we have rich rice germplasm, over 2,000 rice accessions conserved in PGRC Gunnorva. Among that, we selected Masuran variety to test the effect of planting date on flowering time variation and other agronomic traits, because that there is a limited information available on flowering time and other morphological characteristics under different natural environmental conditions. So now I am going to explain about the materials and method. Here we use 2 rice accession, 41, 32, 55, 30 belongs to Masuran variety, and 2 weeks old seedlings are transplanted. The first planting was carried out in November 2018. Likewise, 5 consecutive plantings of 1 month interval were carried out. So data on days to flowering, plant height, total tiller number were recorded at flowering of each planting sets. Then data were analyzed through SPSS software analysis. Here, 2-way ANOVA analysis, 2-factor factorial. Now I am going to discuss about results. The main objective of this study to find out the effect of planting dates on days to flowering, plant height, and total tiller number. In figure 1, explain the effect of planting date on days to flowering in accession number 4132 and 5430. So you can get idea about the flowering time behavior on this graph. In figure 2, explain the effect of planting date on plant height at flowering stage. So there is a significant effect on planting date on plant height at flowering stage, interestingly. And there is a significant interaction effect of plant height, significant interaction effect of planting date and accession on plant height variation and flowering time stage. In figure 3, explain the effect of planting date on total tiller number at flowering time stage. This analysis stated that there is significant interaction effect of planting date and accession on total tiller number variation and flowering time stage. So according to the results, the main objective of this study to find out the effect of planting dates on flowering time variation. So we can get the conclusions through analysis.
Thank you very much. Your time is up. So the paper is open for questions.
Yeah, you can use 1-way ANOVA, no?
No, madam, here we used 2-way ANOVA analysis. Here we consider 2 factors. One is accession, it has 2 levels, and the other one is planting dates, it has 5 levels. So factorial design.
Yeah, you haven't mentioned it here.
In your abstract, actually, you have not mentioned that level of how many levels, how many populations, no? And even significantly, you can't use without p-values. That's statistically, you have to discuss. You can't use this as a normal English term. And what is the number of samples that you have used for each?
10 replicates of each accession.
Okay, right. And here you are, what is the meaning of different planting dates? That means
different planting date means our planting sets planted in 5 consecutive plantings with 1 month interval. It means first accession set planted in November 2018. The other one is December. Likewise, 1 month interval. We are giving 1 month interval for the data.
Okay. Okay. Any questions? Okay, then thank you very much.
Okay, thank you.
Thank you. We have now come to the end of the poster session. Thank you very much to all the poster presenters for the wonderful work done. And we'll be announcing the poster winner for the best poster presentation tomorrow at the end of the technical session. We will now present the token of appreciations to the panel of judges. I cordially invite our track chairperson, Professor Charita Gunasekara on stage to present the tokens. Dr. Himali Jayasingharachi. Dr. Peshala Kumari. Dr. Samira Senanayaka. Thank you, Madam. Thank you. So we have now come to the end of all the sessions. We will see you tomorrow, 8:30 a.m. in the morning, Sri Lankan time. Thank you. Have a good evening.